creature on this planet can match. You think, you remember, you dream. And you may have always assumed, as most people do, that the story of creatures like you, mammals, began sometime after the dinosaurs disappeared, roughly 66 million years ago, when the dust settled and a few small, frightened animals crept out of the rubble to inherit an empty world. That assumption is wrong. Beautifully, magnificently, breathtakingly wrong. The real story of mammals, the real story of you, begins not 66 million years ago, but over 300 million years ago, in a world so ancient and so alien that the very continents looked nothing like they do today. It begins before the first dinosaur ever drew breath. It begins in a period of Earth's history that most people have never heard of, in creatures that most people have never imagined, in an evolutionary drama so vast, so violent, and so extraordinary that the rise of the dinosaurs, for all its fame and cultural glory, is almost a footnote by comparison of the synapsids. This is the story of the lineage that survived everything: volcanic apocalypse, the near total annihilation of complex life, 200 million years of living in the shadow of giants, and persisted, refined itself, and ultimately produced every mammal alive today, including the one watching this video right now. This is your true origin story. Please let us know in the comments where you are watching this history video and what time you are watching it. And be sure to subscribe to the channel for future videos. Let's start at the very beginning, not the beginning of the universe, not even the beginning of life, but the beginning of the chapter that matters most to this particular story. To do that, we need to understand the world as it existed roughly 310 million years ago, deep in a period geologists call the Carboniferous. The Carboniferous world was extraordinary by any measure. The planet was dominated by vast, swampy forests, enormous stands of towering club mosses and tree ferns that stretched across vast stretches of the landmasses. These plants were pumping oxygen into the atmosphere at a rate that the world had never seen before and would not see again, producing an atmosphere so rich in oxygen that insects could grow to staggering sizes. Dragonflies with wingspans of over 2 feet patrolled the skies. Millipedes that stretched nearly 8 feet in length crawled across the forest floors. The air was thick, warm, and extraordinarily productive. It was in this environment, this lush, oxygen-saturated, biologically explosive world, that a critical vertebrate history was quietly taking shape. Vertebrates, creatures with backbones, had already made the long journey from the seas onto land. Fish had developed limbs and the ability to breathe air. The first amphibians had colonized the terrestrial environment, and then, from within that amphibian stock, something remarkable had happened. A group of animals had developed the amniotic egg, an egg with a protective membrane inside that prevented it from drying out, that allowed the embryo to develop in its own private pool of fluid even on dry land. This was revolutionary. This was the key that unlocked the terrestrial world in ways that had never been possible before. The animals that possessed this amniotic egg were called amniotes, and they were the ancestors of everything that would come after: every reptile, every dinosaur, every bird, every mammal. And very early in the history of amniotes, something crucial happened. The amniotes split. They divided into 2 distinct evolutionary branches. This split, one of the most consequential of life on land, occurred roughly 3 million years ago. And understanding it is absolutely essential to understanding everything that follows. On one side of this split were the creatures that would eventually become reptiles and, later, dinosaurs and birds. These are the sauropsids, the lizard-faced ones. Their lineage would go on to produce some of the most iconic animals in the history of the planet. On the other side of this split, and this is the critical point, were the creatures that would eventually become us. These are the synapsids, the fused arch ones, named for a particular feature of their skull structure. And understanding what made synapsids different from the very beginning is the foundation of everything this documentary is about. Because here is the truth that so many people miss: mammals did not evolve from reptiles. This is one of the most persistent misconceptions in popular science, and it deserves to be dismantled clearly and definitively. The synapsid lineage is not a branch that broke off from the reptile family tree. Synapsids and reptiles are siblings, not parent and child. They share a common ancestor, yes, the early amniotes of the Carboniferous, but they diverged from each other over 300 million years ago, and they have been traveling their own separate evolutionary paths ever since. Synapsids did not become reptiles first and then become mammals. They were always something distinct. They were always on the path that would lead, eventually, to warm blood, to live birth, to nursing young with milk, to complex teeth, to large brains, to language, to consciousness itself. Every one of those traits has deep, ancient roots in the synapsid lineage, and those roots were already growing in a world that existed long before the first dinosaur took its first step. This is a story of extraordinary duration. We are talking about a lineage that has been refining itself, adapting, surviving catastrophe, and persisting for over 300 million years. To put that in perspective, the dinosaurs existed as a group for roughly 165 million years. The synapsid lineage has been going for nearly twice that long, and it is still going. Every mammal alive today, from the blue whale to the bumblebee bat, from the elephant to the shrew, is a living testament to this ancient, unbroken thread of survival. So let us go back. Let us go back to the very beginning of the synapsid story, to those ancient carboniferous forests, and let us meet the first members of our extended family. The earliest synapsids looked nothing like mammals. If you could travel back 300 million years and walk through those ancient carboniferous forests, which of course would be an extremely dangerous proposition, you would not immediately recognize any of your distant ancestors. The creatures you would encounter would look superficially quite reptile-like. They were low-slung, sprawling animals, moving on legs that projected outward from their sides rather than directly beneath their bodies. Their scales were rough and protective. Their metabolism was slow and dependent on environmental temperature. In many ways, they would remind you of large, sturdy lizards, but look more carefully. Look at the skull. Even in these earliest synapsids, there is already something different, already a signal of the direction in which this lineage is heading. On each side of the skull, just behind the eye socket, there is a single opening in the bone, a temporal fenestra, scientists call it. This opening, this hole in the skull, is a defining feature of the synapsid lineage. It is the detail that gives them their name, and its significance is enormous. This opening in the skull served a critical function. It provided space for the jaw muscles to expand and contract more powerfully and more efficiently. It allowed for a fundamentally different kind of bite, not just the simple, snapping bite of a lizard, but the beginning of something more complex, more capable, more adaptable. And the jaw, as we will see again and again throughout this documentary, is one of the most important structures in the entire story of mammalian evolution. Changes in the jaw and in the bones associated with the jaw will drive some of the most profound transformations in synapsid history. These early synapsids have been given a somewhat cumbersome scientific name: the pelycosaurs. Though it should be noted that paleontologists today recognize that pelycosaurs are not a natural, unified group. They are more of a grade, a collection of early synapsids that share primitive features but are not all equally closely related to later mammals. Nevertheless, for the purposes of our story, we can use the term to describe the earliest chapter of synapsid evolution, and what a chapter it was. Because almost immediately, on an evolutionary timescale at least, the synapsids began to diversify. They spread across the available habitats of the Carboniferous and into the following period, the Permian. They evolved into a remarkable range of body types and ecological roles. Some were small insectivores, some were large herbivores, some were active, powerful predators, and some of them did something extraordinary. Some of them developed sails. One of the most immediately recognizable early synapsids is Dimetrodon, and if you have ever been to a natural history museum, you have almost certainly seen a model or skeleton of this animal. Dimetrodon is correctly depicted in books and toys as a dinosaur. It is nothing of the sort. It lived 20 to 30 million years before the first dinosaurs evolved. It is more closely related to you than it ever was to Tyrannosaurus rex or Triceratops. Dimetrodon is, in the most genuine sense, a very distant relative of yours. Dimetrodon was an apex predator of the early Permian, a powerfully built animal that could reach lengths of over 11 feet. Its most striking feature was the enormous sail on its back, a fan of elongated spines rising from the vertebrae, connected by a web of skin. This sail has been the subject of considerable scientific debate. Was it used for temperature regulation? Was it a display structure used to attract mates or intimidate rivals? Was it entirely? The honest answer is that we do not know for certain. Different hypotheses have been proposed, and the evidence points in different directions depending on how you interpret it. But what is interesting about Dimetrodon in the context of our story, the sail, is what it represents as a. In the early Permian world, synapsids like Dimetrodon were not marginal creatures, not survivors in ecosystem more successful animals. They were the dominant large terrestrial vertebrates on the planet. They sat at the top of food chains. They were the most ecologically significant large animals on land. This is a fact that cannot be emphasized strongly enough. Dinosaurs existed, it was already successful, dramatically, undeniably, measurably successful. These were the dominant animals of their time. The Permian world was, in a very real sense, a world of synapsids. And that means that when we look back at the history of life on land, we are not looking at a world that was always dominated by reptiles or dinosaurs with mammals as a kind of late arriving afterthought. We are looking at a world that was first dominated by the very lineage that eventually produced us. Alongside Dimetrodon and its carnivorous relatives were an array of other synapsids filling different ecological roles. There were large, heavily built herbivores like Edaphosaurus, another sail-backed synapsid that was feeding on the lush Permian vegetation. There were smaller insectivores and omnivores working the lower levels of the ecosystem. There was an entire web of life built around the synapsid body plan, functioning, evolving, and diversifying across a period of millions of years. And then came the Permian. The Permian period, which ran from approximately 299 million years ago to 252 million years ago, is the critical act in this first chapter of the synapsid story. It is the era in which the synapsid lineage reached its first great peak of diversity and complexity. It is the era in which the most important early steps toward genuine mammalian biology were taken, and it is the era that ended in the single greatest catastrophe in the history of complex life on Earth. To understand the Permian, you first need to understand the world it occupied. The geography of the Permian was dramatically different from the world we know today. The continents were assembled into a single, vast supercontinent called Pangaea, from the Greek for all Earth. This was a colossal landmass stretching from pole to pole, surrounded by a single enormous ocean called Panthalassa. The interior of Pangaea, far from the moderating influence of the surrounding ocean, experienced extreme continental climates, brutal heat in the low latitudes during summer, severe cold in the higher latitudes in winter. Pangaea's geography had profound implications living on it. On one, terrestrial animals could, in theory, disperse across vast distances without having to cross major ocean barriers. Synapsid communities in what is now South Africa were related to and sometimes identical with those in what is now Russia, because the animals could walk between these regions across connected landmasses. On the other hand, the interior of Pangaea was often harsh and arid, subject to monsoon-like seasonal patterns and extended periods of drought. In this challenging world, the synapsids continued to evolve, and during the Permian, they underwent what might be the most important single transformation in their entire evolutionary history. A new group of synapsids emerged, a group that represented a fundamental advance over the earlier, more primitive pelycosaurs. These were the therapsids, and they would go on to produce all subsequent synapsids, including every mammal that has ever lived. The therapsids first appeared in the fossil record in the Middle Permian, roughly 265 million years ago, and from their very first appearance it was clear that something had changed, something significant, something directional. The therapsids were not simply better adapted versions of the older pelycosaurs. They represented a genuine evolutionary shift, a suite of changes that moved the synapsid body plan in a new and consequential direction. It's different. To understand this, we need to think carefully about what a body needs to do in order to be a more effective, more efficient, more capable animal. One of the first things you notice when you look at therapsid skeletons is a change in posture. The old pelycosaurs had that sprawling stance outward from the sides, like a lizard or a crocodile. The therapsids, particularly the more advanced ones, began to bring their legs more directly beneath their bodies. The limbs rotated under the torso, allowing a more upright posture, a longer stride, and a fundamentally more efficient way of moving. Instead of the side-to-side undulation of a sprawling animal, therapsids could move in a straighter line, with less energy wasted in lateral body movements. This change in posture is intimately connected to changes in speed, endurance, and activity levels. An animal with an upright or semi-upright posture can generally move faster and sustain that movement for longer periods than a sprawling animal. The change also affected breathing. A sprawling animal's ability to breathe is compromised when it is moving, because the same side-to-side bending that propels the animal also compresses the lungs alternately. This is one reason why lizards, for all their apparent agility in short bursts, cannot run and breathe efficiently at the same time. The more upright therapsid posture began to separate locomotion from breathing, allowing sustained active movement in a way that was not possible for their predecessors. There were also profound changes in the skull and jaw. The temporal fenestra, that opening in the skull that we noted in the earliest synapsids, enlarged significantly in therapsids. The jaw muscles attached to this opening became larger, more complex, and more powerful. The teeth began to differentiate. In the earliest vertebrates, in fish and in early amphibians, the teeth were essentially all the same, simple conical or peg-like teeth, good for grabbing and holding prey, but not for complex processing of food. Therapsids began to develop what is called heterodonty, different types of teeth for different functions. In the most advanced therapsids, you can see the beginnings of what will become the distinctly mammalian dental pattern: incisors at the front for cutting, large canine teeth for piercing and gripping, and cheek teeth, premolars and molars, for grinding and crushing. This differentiation of teeth is enormously significant. It represents the beginning of food processing that happens in the mouth before swallowing. The ability to chew food thoroughly, to break it down mechanically into smaller pieces, dramatically increases the efficiency of digestion. It allows an animal to extract more energy from the same amount of food in less time. Why does this matter? Because it begins to address one of the most fundamental energetic challenges of the mammalian way of life: the need for a continuous energy input to fuel a warm-blooded metabolism. You cannot continuously generate without a very efficient system for extracting energy. The differentiated teeth and powerful was a critical step blooded lifestyle possible. Speaking of warm blood, one of the most tested questions in all of paleontology: when did synapsids become warm-blooded? When in the long history of this lineage did the transition from ectothermy, relying on external heat sources, to endothermy, generating heat internally, occur? The honest answer is that we do not know precisely. Endothermy does not fossilize. You cannot look at a bone and definitively say, this animal was warm-blooded and this one was not. What you can do is look for proxies, features that are associated with endothermy in living animals, and see when they appear in the fossil record. One such proxy is bone microstructure. The bones of mammals and other endotherms tend to grow way, rapidly, continuously, producing a fibrous high tissue called fibrillation. The bones of ectotherms tend to show slow, cyclical growth patterns, with growth rings analogous to tree rings. When paleontologists began examining therapsid bone microstructure, they found something remarkable. Many therapsids, particularly the more advanced ones, were showing bone growth patterns that looked more like mammals than like lizards. They were growing rapidly. They were growing continuously. Their bones were highly vascularized. These are the signatures of an active, energetically demanding lifestyle. Another proxy is the presence of whisker pits, small openings in the snout bones that, in living mammals, serve as channels for the nerves supplying the vibrancy, the sensitive whiskers. Some therapsid fossils show these openings, suggesting that they may have had some form of hair or whiskers, which would in turn suggest insulation, which would in turn suggest at least some capacity for heat retention, if not full-blown endothermy. The evidence taken together suggests that the transition to endothermy was not a single event, but a gradual process. A slow ratcheting up of metabolic rates and thermogenic capacity across the therapsid lineage over the course of millions of years. It began perhaps facultative capacity, the ability to generate some heat internally in certain circumstances, and gradually became the full-blown, sustained characterizes all living mammals. Some of the most advanced therapsids closest to the mammalian line were partially endothermic. Let us now look more specifically at the therapsid world, at the extraordinary diversity of life that the synapsid lineage produced during the Permian. One of the most spectacularly diverse therapsids were the Dinosaurs. These remarkable animals appeared in the Middle Permian and quickly became one of the dominant groups of terrestrial herbivores on the planet, not just in one region, but virtually everywhere Permian land vertebrates are found. Dinosaurs were in many ways the ecological equivalents of today's large herbivorous mammals, the cattle, the sheep, the wildebeest. They filled the role of bulk plant consumers in Permian ecosystems, processing vegetation in enormous quantities. Dinosaurs had a fascinating and unique body plan, largely toothless. Their gelatinous beak, like they used to crop vegetation, contained two prominent dog teeth. Its name, Dimetrodon, equals two-dog tooth. These large tusk-like structures, particularly in males, and they were likely used in display and combat rather than feeding. The Dinosaurs were extraordinarily successful. They evolved into an enormous range of body sizes, from small, burrowing forms not much larger than a rat to massive, cow-sized, the largest terrestrial vertebrates of the Permian. They lived in forests and open landscapes, in arid environments, and wet ones. Some may have been at least partially aquatic. They occupied virtually every available herbivore niche in the Permian world. Alongside the dicynodonts in the predator guilds of the Permian were the gorgonopsians, and these animals deserve a great deal of attention because they were extraordinary. Gorgonopsians were the apex predators of the middle to late Permian, and they represent a fascinating intermediate state in the evolution of the synapsid body plan, not yet fully mammal-like, but dramatically more advanced than the older pelycosaurs. Imagine an animal the size of a large lion or a polar bear. Now imagine it with a skull full of long, saber-like canine teeth, canines so large that they extended well below the lower jaw even when the mouth was closed. Now imagine it moving with a posture that, while still somewhat sprawling in the hindlimbs, showed a much more upright and efficient stride than any crocodile or lizard. This was a gorgonopsian. This was one of the dominant predators of our Permian world. The largest gorgonopsians, animals like Inostrancevia from what is now Russia, were genuinely formidable creatures, capable of taking down even the largest dicynodont herbivores. Inostrancevia could reach lengths of nearly 12 feet and carried canine teeth that were several inches long. It was without question the apex predator of its ecosystem, the Permian equivalent of a lion, a polar bear, a Tyrannosaurus rex. And all of it, every bit of this predator-prey drama, this ecological complexity, this biological innovation, was happening before the dinosaurs. There was also another critically important group of therapsids in the Permian ecosystem, the cynodonts. And if the gorgonopsians and the dicynodonts are interesting for what they tell us about the diversity of the synapsid lineage, the cynodonts are essential for what they tell us about its direction. Because the cynodonts are the group from which true mammals would eventually descend. The cynodonts, the name means dog tooth, appeared in the late Permian and immediately showed a suite of features that pushed the synapsid body plan significantly further in the mammalian direction. Their teeth were complex and differentiated. Their skulls were highly modified with an enlarged braincase. Their jaws were simplified and more powerful. Some of them show evidence of a secondary palate, a shelf of bones separating the nasal passage from the mouth, which would have allowed them to breathe and chew simultaneously, a critical step toward the sustained activity lifestyle of a mammal. Some cynodonts may have had hair. All of them show skeletal features associated with a more active, more energetic way of life. The cynodonts were not the dominant animals of the late Permian. That role belonged to the gorgonopsians and the dicynodonts. But the cynodonts were there in the ecosystem, beginning the long journey toward mammalhood, and they would turn out to be among the most important animals in the history of life because they were the ones who would survive what was coming. What was coming was the end of the world, not metaphorically, not in an exaggerated, dramatic sense. Literally, the end of the world as the Permian knew it, the most catastrophic event in the history of complex life on Earth. A crisis so severe, so devastating, and so total in its destruction that scientists who study it sometimes struggle to fully convey what it must have been like. The end Permian mass extinction. The Great Dying. 252 million years ago, something happened, several things in fact, all compounding and interacting in catastrophic ways, that killed an estimated 90 to 96% of all marine species and a similarly staggering proportion of terrestrial species. To put those numbers in perspective, the extinction event that killed the dinosaurs 66 million years ago, the one that everyone knows about, the one that has inspired countless books and movies and documentaries, is estimated to have eliminated roughly 75% of all species. That is a catastrophe of unimaginable proportions, but the end Permian event was worse. The end Permian event makes the extinction that killed the dinosaurs look modest by comparison. The primary driver of the end Permian extinction is now well understood, though the details are still being refined by ongoing research. In what is now Siberia, a series of massive volcanic eruptions began, eruptions on a scale that dwarfs anything in recorded human history and most of what the geological record reveals. These eruptions, which geologists call the Siberian traps, were not the explosive, cone-shaped eruptions of a single volcano. They were vast pourings of lava from fissures in the earth, covering an area of roughly 2 million square kilometers, roughly the size of Western Europe, with basaltic rock in some places several kilometers thick. The direct lava flows, however devastating, were not the primary killer. What made the Siberian traps eruptions so catastrophically dangerous was what they put into the atmosphere. The eruptions released enormous quantities of carbon dioxide, methane, and sulfur dioxide. They also burned through massive coal and petroleum deposits in the underlying rock, releasing additional greenhouse gases and toxic chemicals. The carbon dioxide triggered rapid, severe global warming. The sulfur dioxide produced acid rain on a continental scale. The methane potentially released both by the eruptions themselves and by the warming of methane-rich ocean sediments in response to rising temperatures added further warming. The oceans acidified. Ocean oxygen levels plummeted as warming waters held less dissolved oxygen and as the disruption of ocean circulation created vast dead zones. The land was scorched by heat and drenched with acid. The speed of this environmental collapse is one of the things that makes it so catastrophic. The evidence suggests that much of the most severe damage happened in a geologically brief interval, perhaps as short as 60,000 years, possibly even less in the most acute phase. For complex ecosystems that had been building for millions of years, this was essentially instantaneous. There was no time to adapt, there was no time to migrate to safer regions. The world simply changed faster than life could respond. In the oceans, entire groups vanished forever. The Rugose corals, which had built reef systems across the Paleozoic seas for hundreds of millions of years, disappeared completely. The tabulate corals were gone. Many groups of brachiopods, mollusks, echinoderms, major components of Paleozoic marine ecosystems, were either completely eliminated or reduced to tiny remnant populations. The trilobites, those iconic Paleozoic arthropods that had existed for over 250 million years, were finally definitively extinguished. The sea simply died. On land, the destruction was equally severe. The Permian terrestrial ecosystems, those diverse communities of synapsids, early reptiles, amphibians, and plants that we have been exploring, collapsed catastrophically. The lush forests of the Permian were devastated. Large areas of land may have been effectively stripped of vegetation, leaving bare soil exposed to erosion. The food chains that had supported the dicynodonts, the gorgonopsians, the cynodonts, and all the other Permian animals collapsed from the bottom up. For the synapsids, the effect was nearly annihilating. The gorgonopsians, those magnificent saber-toothed apex predators that had been among the dominant terrestrial carnivores of the late Permian, were completely wiped out. Every last one. Not a single gorgonopsian survived the end Permian extinction. The entire group simply ceased to exist. Most of the dicynodonts, the wonderfully diverse, globally distributed herbivores that had been so successful throughout the Permian, were gone as well. Only one or possibly a few lineages scraped through. The extraordinary diversity that had characterized the group in the Permian was replaced by a handful of surviving forms. Other therapsid groups, the dinosaurs, the Biormasuchia, others, were completely eliminated. The synapsid lineage, which had been the dominant force in terrestrial vertebrate life for tens of millions of years, was reduced to a handful of survivors clinging to existence in a devastated world. And among those survivors were the cynodonts. Why the cynodonts? Why did this particular group make it through when so many others did not? There is no single definitive answer. Extinction events rarely leave us with clean explanations for every case of survival and extinction. But we can identify features of the cynodont body plan that were likely advantageous in the post-extinction world. Size matters in an extinction event. Small animals generally fare better than large ones during crises because they require less food, can exploit a wider range of food sources, and can hide more effectively from environmental extremes. The cynodonts of the late Permian were not, in general, large animals. They were mostly small to medium-sized, roughly in the range of a cat to a dog. This small size would have helped them survive when food was scarce and conditions were extreme. Their physiology likely helped as well. If the cynodonts were, as the evidence suggests, moving in the direction of endothermy, generating some of their own body heat internally, they would have been somewhat less vulnerable to temperature swings than a fully ectothermic animal. They could keep their bodies functioning at least partially, regardless of ambient temperature. This would have been valuable in a world where temperatures were fluctuating wildly. Their diverse, complex teeth would have given them dietary flexibility. An animal that can eat a wide range of food types, insects, seeds, small vertebrates, plant material, has a much better chance of survival in an ecosystem that has been largely destroyed than an animal that depends on a specific type of food that may no longer be available. And there may have been an element of sheer luck. Extinction events are not perfectly selective. They do not kill only the weak animals and spare the strong. There is a random component to survival, particularly when the crisis is as severe and as rapid as the end Permian event. Some lineages survive because their members happen to be in the right place at the right time, in refugia where conditions were somewhat less extreme, where food was somewhat more available, where the worst of the environmental chaos was somewhat moderated. Whatever the combination of factors, the cynodonts made it through. And in making it through, in scraping past the most devastating event in the history of complex life on Earth, they preserved the lineage that would eventually produce every mammal on the planet. This is the first and most profound bottleneck in our evolutionary history. The first time this lineage almost ceased to exist entirely. The first near miss in a story that would have several more. And the fact that we are here today, the fact that you are watching this video right now, is, in the most direct possible sense, a consequence of the fact that some small, tough, metabolically active, dietary flexible cynodonts found enough food and enough shelter to survive in the wreckage of the Permian world. Think about what that means. Every human being who has ever lived, every mammal that has ever drawn breath in the last 252 million years. All of it, all of that staggering diversity and biological richness, all of it traces back to a handful of small animals that survived in what must have been a devastated, dying world. The world that emerged from the end Permian extinction was profoundly different from the one that had existed before. It took life an estimated 5 to 10 million years to fully recover from the crisis, to rebuild the ecological complexity, the biodiversity, the stable food chains that characterize a fully functioning biosphere. In the meantime, the survivors lived in a simplified, stressed, ecologically impoverished world, but life, as it always does, began to rebuild. The period immediately following the end Permian extinction is called the early Triassic, and it was a world in transition. It was a world where the old order had been demolished and new animals were beginning to fill the ecological vacancies left by extinction. It was a world of opportunity, brutal, harsh opportunity in a degraded environment, but opportunity nonetheless. And in this early Triassic world, something remarkable happened. The synapsids, reduced, battered, barely surviving, showed a brief resurgence. The Dicenodont survivors in particular recovered quickly, producing a new radiation of forms that, for a few million years, reasserted synapsid dominance in terrestrial ecosystems. Animals like Lystrosaurus, a heavily built, beak-mouthed Dicenodont about the size of a large pig, became extraordinarily common in the early Triassic, so common that their fossils appear in huge numbers in rocks of this age from multiple continents. Lystrosaurus was, for a time, among the large terrestrial vertebrates on the planet. It was in its own way a success story, a testament to the ability of the synapsid lineage to recover and rebuild. But the synapsids would not retain their dominance for long, because in the Triassic, the world was changing again. New groups of animals were evolving, diversifying, and moving into the ecological spaces that had been cleared by the extinction event. Among these new animals were the archosaurs, the group that would eventually give rise to the dinosaurs, the pterosaurs, and ultimately the birds. The archosaurs, and specifically the dinosaur lineage within the archosaurs, proved to be formidable competitors. Through the Middle and Late Triassic, as the world recovered from the end Permian extinction, the dinosaurs underwent a rapid diversification. They evolved into a remarkable range of body types and ecological roles. They developed upright postures, efficient bipedal locomotion, high metabolic rates. They competed with the synapsids for ecological space, and in this competition, for reasons that paleontologists are still debating, the dinosaurs won. Or more precisely, the dinosaurs came to dominate the large-bodied terrestrial vertebrate niches. They became the big animals, the large herbivores, the large carnivores, the dominant ecological players. And the synapsids, the cynodonts in particular, were pushed into smaller niches, smaller body sizes, more specialized and marginal ecological roles. There is a second extinction event that may have played a role in this transition. At the end of the Triassic, roughly 201 million years ago, another mass extinction occurred, the end Triassic event. This extinction also associated with massive volcanic eruptions, this time in what is now the Central Atlantic magmatic province, further reduced synapsid diversity and opened ecological space that the dinosaurs were well positioned to exploit. By the early Jurassic, the pattern that would define the next 135 million years was firmly established. The dinosaurs were the dominant large terrestrial vertebrates. The synapsids had been reduced to small, mostly nocturnal forms living in the ecological margins, in the undergrowth, in burrows, in the dark hours of the night. But what had happened exactly to the synapsids during the Triassic? What had the cynodont lineage been doing as the dinosaurs rose to dominance? They had been evolving, quietly, persistently, in ways that would prove enormously consequential. While the big dinosaurs were claiming the large animal niches, the cynodonts were refining the features that would define the mammal body plan. And during the Triassic, the most important transition in all of synapsid history occurred. True mammals evolved. The definition of true mammal is itself a subject of scientific debate, because the transition from cynodont to mammal was not a single, decisive event, but a gradual process. There was no moment in the Triassic when a cynodont gave birth to a true mammal. evolution does not work that way. Instead, the mammalian features accumulated gradually, generation by generation, over millions of years. But there is a convenient and widely used marker that paleontologists use to define the mammalian grade, the condition of the jaw joint. In all living reptiles, the jaw is articulated. The lower jaw connects to the skull through two bones called the quadrate and the articular. In all living mammals, the jaw joint is formed by two completely different bones: the dentary, the main bone of the lower jaw, and the squamosal, a bone of the skull. The quadrate and articular have not disappeared, they have migrated. In mammals, these bones have moved from the jaw joint to the middle ear, where they function as the malleus and incus, two of the three tiny bones that transmit sound vibrations in the mammalian ear. This transformation, the migration of jaw bones into the ear, is one of the most extraordinary things that happens anywhere in the fossil record. It is a transformation that, if you described it to someone without the fossil evidence, would seem almost impossible. How could jaw bones become ear bones? How could the joint between the upper and lower jaw migrate into the skull to become part of the hearing apparatus? The answer is that it happened gradually, step by small step, over millions of years. And the fossil record of therapsids and early mammals actually documents this transition beautifully. In the most primitive cynodonts, the dentary bone of the lower jaw is just one of several bones in the lower jaw, and the jaw joint is the standard quadrate articular type. In progressively more derived cynodonts, the dentary becomes larger and larger, and the other bones become smaller and smaller. In the most advanced cynodonts, the ones closest to the mammalian line, you have a jaw where the dentary is enormous and dominant, and the other bones are tiny slivers of bone at the back of the jaw, still connecting the jaw to the skull but barely. The transition is visible. You can see it happening, and in the earliest true mammals, those tiny bones have completed their migration. They have become part of the ear. The jaw joint is now a dentary squamosal joint. And the result is a hearing apparatus of extraordinary sensitivity, three bones instead of one transmitting vibrations from the eardrum to the inner ear, providing a mechanical advantage and a precision that far exceeds the hearing of any reptile. Why did this happen? Why did jaw bones become ear bones? This is a question that has fascinated evolutionary biologists for decades. The most persuasive explanation involves the interaction between jaw mechanics and hearing, and the way that selection pressures can cause a structure to be modified for one function when it is already partially serving another. In advanced cynodonts, the non-dentary bones at the back of the jaw were so small that they were likely no longer making a major contribution to jaw strength. They were, in a sense, already becoming redundant in their original role. But they were still in contact with the skull, and they were probably transmitting some vibrations to the inner ear, functioning, in a crude way, as part of the hearing apparatus even while still technically being part of the jaw. Once natural selection recognized the auditory value of these bones, once even a slightly improved version of this arrangement provided any advantage in detecting sounds, the evolutionary trajectory was set. Generation by generation, the bones were refined for their new auditory role rather than their old jaw role, until the transition was complete. The result was an ear of exceptional sensitivity, the mammalian middle ear with its three tiny ossicles, capable of detecting a vastly broader range of frequencies and a much greater range of sound intensities than the simpler ear of a reptile. This enhanced hearing would become one of the most important tools in the mammalian survival toolkit during the age of dinosaurs, as we will see in the second part of our story. Let us pause here, in the late Triassic, and take stock of where we are and what we have covered. We began over 300 million years ago in the Carboniferous period, where the first amniotes were splitting into two lineages, the sauropsids that would eventually become reptiles and birds, and the synapsids that would eventually become us. We watched the synapsids diversify into the pelycosaurs and then the therapsids, becoming the dominant terrestrial vertebrates of the Permian. We encountered the extraordinary diversity of the Permian synapsid world, the dicynodonts, the gorgonopsians, the early cynodonts, and marveled at the richness and complexity of an era that most people have never heard of. We then witnessed the catastrophe of the end Permian extinction, the near total annihilation of complex life on Earth, and watched the synapsid lineage nearly perish, reduced from a position of dominance to a handful of desperate survivors. We followed those survivors, the cynodonts, through the Triassic, watching them lose the competitive battle with the rising dinosaurs and retreat into small body niches, while simultaneously undergoing the most important transformation in their evolutionary history, the transition to true mammals. In the late Triassic and early Jurassic, the first true mammals were emerging small, probably nocturnal, certainly active and warm-blooded, with complex teeth and sensitive ears and large brains relative to their body size. These were animals the size of mice and shrews, living in a world increasingly dominated by dinosaurs. They were by any conventional measure a minor component of the world's ecosystems, small, marginal, easily overlooked, but they were there. They were surviving. They were diversifying in their own quiet way. They were accumulating the biological innovations that would, tens of millions of years in the future, allow their descendants to inherit the world. The first true mammals, the animals that mark the culmination of everything we have been discussing, the end product of over 60 million years of synapsid evolution from the earliest pelycosaurs to the first mammals, appeared in the late Triassic, roughly 225 to 210 million years ago. They were tiny. Most were probably no larger than a small mouse. Their fossils are often nothing more than small fragments of jaw bone with a few teeth attached. But those teeth tell an extraordinary story. Mammalian teeth are among the most informative structures in the entire fossil record, because mammals, uniquely among vertebrates, replace their teeth only once in their lifetime, from milk teeth to adult teeth, and then must use those adult teeth for the rest of their lives. The teeth have been under intense selective pressure to be durable, precise, and effective. Mammalian teeth have evolved into complex structures with precise interlocking cusps and ridges, structures that must fit together exactly with the corresponding teeth on the opposite jaw in order to function efficiently. This precision of tooth occlusion, the way upper and lower teeth mesh together, is uniquely mammalian. No reptile, no other vertebrate group has anything like it. And it requires a very specific constraint. The teeth cannot continue to be replaced throughout life, because each new set of teeth would have to be precisely matched to the opposing set, and the complexity of the matching makes continuous replacement impractical. So mammals evolved a system of just two sets of teeth, milk teeth and adult teeth, and relied on the durability and precision of those adult teeth for their entire lives. The earliest mammals' teeth show this pattern in an early, developing form. They show the beginnings of the complex cusps and ridges that will characterize mammalian teeth throughout their subsequent history. And they show something else important. They show dietary specialization that suggests these early mammals were not generalist feeders but were already adapting to specific food sources, primarily insects and other invertebrates, which were probably the most reliable food source available in the ecological margins they inhabited. Who were these first mammals? What did they look like? How did they live? These are questions that paleontologists have been working to answer for decades, and the picture that has emerged is both detailed and fascinating. While much remains uncertain, soft tissue, behavior, and physiology rarely fossilize, the combination of fossil bones, comparative anatomy, and modern imaging technology has given us a surprisingly rich portrait of early mammalian life. In body form, the earliest mammals were probably somewhat shrew-like, small, compact bodies, relatively short legs, long snouts equipped with sensitive whiskers. They almost certainly had hair. The insulation that hair provides would have been essential for maintaining body temperature in small, warm-blooded animals, because small bodies lose heat much more rapidly than large ones, a consequence of their high surface area to volume ratio. They were almost certainly warm-blooded in the full mammalian sense, maintaining a stable, elevated body temperature through internal metabolic heat generation. Their eyes were almost certainly large relative to their skull size, another adaptation to nocturnality. If you have to navigate the world in darkness, you need to collect as much light as possible, which means large eyes. This adaptation to nocturnal life will leave profound traces throughout the early history of mammals, influencing not just their visual systems, but many aspects of their behavior, physiology, and neurology. Their brains were large relative to their body size. Already in these earliest true mammals, the trend toward neural complexity that will become one of the defining features of the mammalian lineage was beginning to express itself. The olfactory bulbs, the brain regions devoted to the sense of smell, were particularly well developed, reflecting the importance of scent in a nocturnal world where visual information is limited. The cerebral cortex, the brain region associated with complex cognition, was beginning to expand. The reproductive biology of the earliest mammals is a subject of ongoing debate and investigation. The living mammals are divided into three groups based on their reproductive strategies: the monotremes, platypus, and echidnas, which lay eggs; the marsupials, which give birth to very underdeveloped young that continue development externally; and the placental mammals, which nourish their offspring internally via placenta for an extended period. The question of which of these reproductive strategies was ancestral, which came first, has been debated for many years. The current evidence suggests that the egg-laying strategy of monotremes is likely the most primitive, the most similar to the ancestral mammalian condition. The earliest true mammals were probably egg-layers like the platypus. They would have laid small, leathery eggs, not the hard-shelled eggs of a bird, but soft-shelled eggs more like those of a reptile, and the hatchlings would have been tiny and altricial, requiring considerable parental care. They were probably nursed with some form of nutritious secretion from the mother's skin, the evolutionary precursor of milk, which would have supplemented the food that the hatchlings could obtain on their own. This system of parental investment, of producing small numbers of offspring and investing heavily in their development and survival, is a critical mammalian innovation. It stands in stark contrast to the strategy of most reptiles, which produce large numbers of eggs and provide little or no parental care, relying on sheer numbers to ensure that at least some offspring survive. The mammalian approach, few offspring, intense parental investment, is a bet on quality over quantity. It is a strategy that pays off when offspring need extended learning and development in order to function effectively in a complex world. The earliest mammals were also almost certainly solitary or lived in very small groups. The combination of territorial behavior, cryptic lifestyle, and small body size makes complex social organization unlikely in the earliest forms. But parental care, particularly maternal care, was probably already well developed. The helpless, dependent young of an egg-laying mammal require substantial investment from the mother, and this investment, this early bond between mother and offspring, may be the evolutionary root of the complex social bonding that characterizes so many mammalian groups today. Now, as we approach the end of the first half of our story, let us take a broader view and consider what all of this means, what the lessons of these first 300 million years of synapsid history tell us about the nature of evolution and survival. The first thing that stands out is persistence. The synapsid lineage is extraordinarily old. It is older than the dinosaurs. It has survived multiple major extinction events. It has spent extended periods as a marginal, minor component of ecosystems dominated by other animals. And yet it has persisted quietly, tenaciously, continuously refining itself, and ultimately produced the most complex animals that have ever lived. This is not the story that most people have in their heads when they think about evolution. The popular conception of evolution is often one of linear progress, simple to complex, primitive to advanced, fish to amphibian to reptile to mammal to human, each stage replacing the previous one as it becomes obsolete. But the real story is far more complicated and far more interesting than that. The synapsids did not replace the reptiles. They evolved in parallel with them, diverging from a common ancestor over 300 million years ago. The synapsids were not always the winners. They lost the competitive battle with the dinosaurs in the Triassic and spent the entire Mesozoic era as marginal animals. And yet they survived, they endured, they persisted through catastrophes that destroyed the dominant groups of their time. The second lesson is the importance of seemingly minor innovations. The temporal fenestra in the synapsid skull, that small hole in the bone behind the eye socket, was a seemingly modest change. But it allowed for improved jaw musculature, which enabled more complex teeth, which made possible more efficient digestion, which powered a higher metabolic rate, which ultimately drove the transition to warm blood. One small structural change cascaded over millions of years into the entire mammalian way of life. Similarly, the bones at the back of the cynodont jaw, bones that were already becoming redundant in their original role, were repurposed into the most sensitive hearing apparatus in the vertebrate world. A constraint became an opportunity. A vestige became an innovation. This is how evolution actually works, not by grand sweeping design, but by the accumulation of small incremental changes, each building on what came before, each constrained by what already exists, but each capable of being co-opted, modified, and repurposed in ways that are not predictable in advance. The third lesson, and perhaps the most humbling, is contingency. The history of life on Earth could have gone very differently. If the cynodonts had not survived the end Permian extinction, there would be no mammals. No whales, no elephants, no bats, no primates, no humans. The world today would be utterly different, probably dominated by the descendants of whatever archosaur groups had survived in a biological landscape that we cannot even clearly imagine. The fact that we exist, the fact that any mammal exists, is a consequence of a sequence of contingent events, each of which could have gone another way. The cynodonts survived the end Permian extinction. The early mammals survived the end Triassic extinction. A small population of some early primate ancestors survived the pressures that drove most of the large mammals to extinction in the late Pleistocene. and so on back through the chain of causation, each link dependent on the one before. We are here by a kind of cosmic accident, not random exactly, but deeply contingent, not inevitable. Not the foreordained outcome of some progressive march toward complexity. We are the consequence of a pre-dinosaur experiment that almost ended multiple times before it could produce its most complex results. Let us now look carefully at a few specific animals that illuminate this first phase of our story, animals that deserve more attention than the mere passing mentions they typically receive in popular accounts of prehistoric life. Consider Progynopsis, one of the earliest cynodonts from the late Permian of what is now South Africa and parts of Germany. This animal was roughly the size of a small dog. It had a long snout and a complex, differentiated set of teeth. Its skeleton shows a mosaic of ancestral and derived features, still somewhat sprawling in posture, but with limb proportions and joint configurations that suggest it was capable of a more efficient, more active style of locomotion than the earlier therapsids. Progynopsis may have been at least partially aquatic. Some features of its skeleton suggest adaptations for swimming, which would make it a unique and fascinating character in our story. It was swimming in ancient rivers and lakes while the great Gorgonopsians still hunted the plains above. Or consider Thrinaxodon, a slightly younger and more derived cynodont from the early Triassic, known from beautifully preserved fossils from what is now South Africa and Antarctica. Thrinaxodon is particularly fascinating because of what its fossils reveal about its behavior. Some Thrinaxodon fossils have been found in burrows, actual preserved burrow structures in the rock, suggesting that these animals were using burrows for shelter, for protection from environmental extremes, possibly for rearing young. And in at least one extraordinary case, a Thrinaxodon has been found sharing a burrow with a temnospondyle amphibian, preserved together in what appears to be evidence of shared shelter, perhaps both animals using the same burrow for protection during a difficult period. Burrow-dwelling behavior would have been enormously advantageous during the harsh conditions of the post-extinction early Triassic world. A burrow provides shelter from temperature extremes, protection from predators, and a stable microenvironment in which the animal's thermoregulatory demands are reduced. And the fact that Thrinaxodon was burrowing suggests a level of behavioral complexity and environmental awareness that goes beyond simple reflex responses to immediate stimuli. Consider also Morganeucodon, one of the earliest true mammals, from the late Triassic and early Jurassic of what is now Wales, China, and several other locations. Morganeucodon was tiny, barely larger than a mouse, and is known from fragmentary but informative fossils. Its teeth show the characteristic mammalian pattern of precise occlusion, with complex cusps interlocking precisely between upper and lower jaws. Its jaw shows the transitional condition between the cynodont and mammalian states, with a large dentary and several small accessory jaw bones still present at the back. Its ear region suggests the beginnings of the improved mammalian hearing system. Morganeucodon was not yet quite a real mammal in the strict anatomical sense. Its jaw joint was still partially the old quadrate articular type, with the dentary squamosal joint developing alongside it. But it was right at the threshold, right at the cusp of the transition. It represents a moment frozen in time, the evolutionary frontier between cynodont and mammal, the moment when the most consequential transformation in synapsid history was reaching its completion. There is one more thing that deserves extended discussion before we close the first half of our story, and it concerns the Permian period specifically, the great synapsid world before the end Permian extinction. Because this world is so poorly known to the general public, and because it is so important to the story we are telling, it deserves more than a brief overview. The Permian synapsid fauna, particularly the late Permian fauna of the Karoo Basin in what is now South Africa, is one of the most remarkable and informative collections of prehistoric animals known to science. The Karoo Basin contains a nearly continuous fossil record of Permian and early Triassic vertebrates that allows paleontologists to track evolutionary change, ecological structure, and the effects of the end Permian extinction in extraordinary detail. What the Karoo record shows is a world of extraordinary ecological complexity, a world that, while superficially unfamiliar, operated on principles that are entirely recognizable. There were apex predators, the Gorgonopsians, controlling prey populations. There were large herbivores, the Dicynodonts, processing plant material in bulk. There were smaller omnivores and insectivores, the smaller cynodonts and other therapsids, filling the intermediate roles. There were ecological relationships between predator and prey, between herbivore and plant, between competitor and competitor. that mirror in broad outline the ecological dynamics of any modern terrestrial ecosystem. This is important because it challenges a persistent narrative that frames the Permian as somehow primitive or transitional, as merely the prologue to the more familiar Mesozoic world. It was not a prologue. It was a fully realized, mature, ecologically complex world in its own right, populated by animals with their own adaptations, their own behaviors, their own evolutionary histories. The Karoo Gorgonopsians were not proto-predators stumbling toward a more effective body plan. They were effective, successful predators that had been refined over millions of years. In Austransevia, hunting through what is now Russia was as complete and effective a predator in its world as any lion or wolf is in the modern world. The Dicynodonts browsing across the Karoo landscapes were not primitive, inefficient herbivores. They were successful, widespread, and ecologically crucial components of a functioning ecosystem. And then the Permian extinction ended it swiftly, catastrophically, nearly completely. The Karoo record documents this with heartbreaking clarity, richly diverse faunas below a certain geological horizon, and then above that horizon the impoverished, simplified fauna of the early Triassic, with Lystrosaurus dominating a devastated world. One final thought before we close the first part of our story. There is something profound and in its own way deeply moving about what the synapsid story tells us about the nature of life and time. We live in an age obsessed with the immediate, with the present, with the recent past, with the near future. The time scales of geology and evolutionary biology are so vast, so utterly beyond ordinary human experience that they can be difficult to make emotionally real. But consider, when Dimetrodon was hunting through the forests and plains of the early Permian, the dinosaurs were not yet a thought in evolution's eye. They would not exist for another 40 million years. The entire span of the dinosaurs' existence, 165 million years of the most spectacular large animals that ever walked the earth, had not yet happened. And yet the synapsid lineage was already there, already thriving, already accumulating the adaptations and innovations that would eventually, after surviving the greatest catastrophe in the history of complex life, after spending over 130 million years in the margins of a world dominated by dinosaurs, produce the most complex minds that evolution has ever fashioned. We are old, far, far older than we typically imagine. Our roots do not go back to the Cretaceous, to the age of Triceratops and Tyrannosaurus. They go back to the Permian. To the Carboniferous, to a world before the dinosaurs were born. We carry within us the biological inheritance of over 300 million years of evolution, the teeth, the ears, the brain, the warm blood, all of it refined across an inconceivable expanse of time. The dinosaurs were spectacular, but the synapsid story is older, longer, and ultimately more consequential. It is the story that led to minds capable of asking the question, where did we come from? And now we begin to find out. In the second part of our story, we will follow the early mammals through their long Mesozoic odyssey. 165 million years of living in the shadow of the dinosaurs, diversifying in ways that the fossil record has only recently begun to reveal in detail, refining the biological innovations that would eventually allow mammals to take over the world. We will explore the extraordinary diversity of Mesozoic mammals that has been uncovered in recent decades, the gliding forms, the swimming forms, the burrowing forms, the larger predators. We will examine the crucial role of the nocturnal lifestyle in shaping mammalian sensory biology, and we will witness the final catastrophe, the end Cretaceous extinction that cleared the stage for the mammalian revolution. The story of our origins is not yet half told. Stay with us. We left our story at a moment of profound transition. The first true mammals had emerged from the cynodont lineage in the late Triassic, tiny and secretive, warm-blooded and sharp-eared, carrying within their small bodies the accumulated biological inheritance of over 60 million years of synapsid evolution. They had survived the end Permian extinction, the most devastating catastrophe in the history of complex life, and they had watched from the ecological margins as the dinosaurs rose to dominance and claimed the large animal niches of the terrestrial world. They were small, they were largely nocturnal. They were, by any conventional measure of ecological success, minor players in a world dominated by giants, and yet they were there. They were there in the Triassic, and they were there in the Jurassic, and they were there in the Cretaceous. For 165 million years, a span of time so vast that it exceeds the entire duration of the dinosaurs' existence, the mammals persisted alongside the dinosaurs, living in the cracks and margins of the Mesozoic world, refining their biology, diversifying their body plans, sharpening their senses, expanding their brains. They did not merely survive the age of dinosaurs. They evolved through it, changed through it, became through it the animals that would eventually inherit the world. This second part of our story covers that long Mesozoic odyssey, and it is a story that has been revolutionized in the past 3 decades by an extraordinary wave of fossil discoveries that have completely overturned the old, simplistic picture of Mesozoic mammals as uniformly tiny, uniformly shrew-like creatures passively waiting for the dinosaurs to die. The truth, as the fossils now reveal, is far more complex, far more dynamic, and far more surprising than anyone imagined even 30 years ago. But before we dive into that new picture, we need to understand the world in which the early mammals found themselves, the Mesozoic world, the world of the dinosaurs, and what it meant to be a small, warm-blooded animal living in the shadow of the largest terrestrial animals that ever walked the earth. Please let us know in the comments where you are watching this history video and what time you are watching it, and be sure to subscribe to the channel for future videos. The Mesozoic era, the age of dinosaurs, is traditionally divided into 3 periods: the Triassic, the Jurassic, and the Cretaceous. Together, these 3 periods span approximately 186 million years, from roughly 252 million years ago to 66 million years ago. It is, by any measure, an enormous span of time, long enough for the Atlantic Ocean to open from nothing to a substantial body of water, long enough for the continents to drift thousands of miles from their Triassic positions, long enough for mountains to rise and erode and rise again. During this immense span of time, the dinosaurs were the dominant large terrestrial vertebrates on land. They evolved into an extraordinary diversity of forms, from the small, swift, bipedal theropods to the enormous long-necked sauropods that are among the largest animals that have ever lived, from the armored ankylosaurs to the horned ceratopsians, from the bone-headed pachycephalosaurs to the duck-billed hadrosaurs. Every large animal ecological role on land with an inventiveness and variety that still astonishes the scientists who study them. And through all of it, through the entire, magnificent 165 million year spectacle of the dinosaurs, the mammals were there, watching, surviving, in the trees, in the burrows, in the leaf litter, in the darkness of the night, small, warm, quick, sharp-sensed, and persistent. The relationship between the mammals and the dinosaurs during the Mesozoic was not simply one of oppressor and oppressed, of giant dominating tiny. It was more complex than that. The dinosaurs did not specifically hunt mammals. Most of them were far too large to bother with such small prey, and those that were the right size to prey on small mammals were doing so in the context of a much larger ecological picture. The mammals were not primarily constrained by direct predation from dinosaurs. They were constrained by competitive exclusion, by the simple fact that the dinosaurs occupied the large body ecological niches so thoroughly and so effectively that there was no room for mammals to grow large and diversify into those roles. This competitive exclusion was the primary driver of the mammalian nocturnal lifestyle. The dinosaurs were day animals, or at least, the evidence suggests that most dinosaurs were active during daylight hours. The mammals, by retreating to the night, were exploiting a temporal niche that the dinosaurs largely left open. The night was theirs, and in claiming the night, the early mammals set themselves on an evolutionary trajectory that would profoundly shape the biology of every mammal alive today. The nocturnal lifestyle imposes specific demands on an animal's sensory biology. In the dark, vision is limited. The specific wavelengths of light that color vision depends on are simply not available in sufficient quantities. An animal that wants to function effectively at night needs to either develop extremely sensitive light-gathering eyes or compensate for reduced visual acuity by developing other senses to a high degree, or both. The early mammals did both, and the consequences of this sensory adaptation are still written clearly in the biology of every living mammal, including humans. Consider vision first. The eyes of early mammals underwent significant reorganization in response to nocturnal selection pressures. The retina, the light-sensitive layer at the back of the eye, contains 2 types of photoreceptor cells: rods, which are sensitive to low levels of light but do not distinguish colors, and cones, which require brighter light but are responsible for color discrimination. A diurnal animal, one active during the day, typically has a retina rich in cones, allowing fine color discrimination. A nocturnal animal needs rods far more than cones. The early mammals appear to have undergone a dramatic reorganization of their retinas, reducing the number and types of cone cells and increasing the proportion of rod cells. This reorganization was so thorough that it left a permanent mark on the mammalian lineage. Even today, most mammals have relatively poor color vision compared to, say, birds or many reptiles, which are, of course, the descendants of the diurnal dinosaurs. Birds can see into the ultraviolet, Many lizards have 4 or even 5 types of cone cells. Humans, like most primates, have 3 types of cone cells, and we have 3 only because the primate lineage later re-evolved a third cone type after the nocturnal bottleneck had reduced our ancestors to 2. Many other mammals, dogs, cats, most rodents, still have only 2 types of cone cells, a direct consequence of the nocturnal lifestyle of the early mammals. This is called the nocturnal bottleneck, the period in early mammalian evolution during which the predominantly nocturnal lifestyle of the mammals effectively stripped away much of the color vision capacity that the ancestral amniotes had possessed. It is one of the clearest examples anywhere in vertebrate biology of how a specific ecological context can drive permanent changes in sensory biology, changes so deep and so fundamental that they persist for hundreds of millions of years afterward. But while color vision was being reduced, other senses were being dramatically enhanced. The sense of smell, olfaction, became enormously important in the nocturnal mammalian world. Smell does not depend on light. Smell carries information about food, predators, mates, and territory even in total darkness. And the early mammals invested heavily in olfactory capacity. The evidence of this investment is visible in the fossil record. The skulls of early mammals show a dramatically enlarged olfactory region. The nasal cavity was complex and extensive, with the bony scrolls called turbinates that increased the surface area available for olfactory epithelium. The olfactory bulbs of the brain, the regions devoted to processing smell information, were large relative to overall brain size. The mammalian sense of smell was already, in these early forms, far more sensitive and discriminating than that of any reptile. The genetic evidence confirms this picture beautifully. The mammalian genome contains an enormous number of genes dedicated to olfactory receptors, the proteins on the surface of nasal cells that bind to specific odor molecules and trigger nerve signals. Humans, despite having lost the use of many of these genes over our evolutionary history, as vision became more important in our primate ancestors, still carry roughly 800 olfactory receptor genes in our genome. Dogs have over 1,000, mice have even more. The mammalian nose is a biological instrument of extraordinary sophistication, and its sophistication traces directly to the demands of life in the dark world of the early Mesozoic. The sense of hearing was also profoundly enhanced during the early mammalian period. We discussed in Part 1 the extraordinary transformation by which jaw bones migrated to become ear bones, the evolution of the mammalian middle ear with its 3 ossicles. This transformation was already complete in the earliest true mammals, and its auditory consequences were profound. The 3-ossicle mammalian middle ear is, by a significant margin, the most sensitive hearing apparatus in the vertebrate world. It can detect a vastly wider range of frequencies than the simpler hearing systems of reptiles or amphibians, and it can respond to sound intensities far lower than those detectable by any non-mammalian ear. For an animal living in the dark, navigating by sound, detecting the movements of prey and predators in the leaf litter and the undergrowth, this enhanced hearing was an enormous advantage. Early mammals could likely hear the high-frequency sounds produced by insects, sounds that most reptiles cannot detect at all. They could hear the footsteps of predators approaching through the undergrowth. They could communicate with each other and with their offspring using sounds that were inaudible to the dinosaurs around them, a private channel of communication, invisible to the dominant animals of the day. The expansion of brain tissue associated with these enhanced sensory systems was also significant. The brain of a mammal is not simply a scaled-up reptile brain. It is a fundamentally reorganized brain, with regions devoted to olfactory and auditory processing that are proportionally much larger than in any reptile, and with an expanded neocortex, the outer layer of the cerebral hemispheres, that is the seat of complex sensory integration, learning, memory, and in its most elaborate forms, consciousness and abstract thought. The mammalian neocortex is unique in the vertebrate world. No other group of animals has anything quite like it. And its origins lie precisely in the demands of the nocturnal lifestyle of the early Mesozoic mammals, the need to integrate complex olfactory, auditory, and tactile information in real time, in darkness, in order to navigate a world where a mistake could be fatal. Now let us turn to the fossils themselves, to the remarkable evidence that has been uncovered over the past several decades that has transformed our understanding of Mesozoic mammalian diversity. The old picture of Mesozoic mammals was bleak and monotonous. For most of the 20th century, the dominant view was that Mesozoic mammals were essentially all the same, small, shrew-like insectivores, uniform in their tiny body size, uniform in their nocturnal lifestyle, ecologically static through the entire 165 million years of the dinosaurs' reign. They were seen as animals in a holding pattern, maintaining their basic body plan unchanged while waiting for the dinosaurs to clear the stage. This picture has been shattered, utterly and irreversibly shattered by a flood of new fossil discoveries, primarily from China but also from other locations around the world, that has revealed a Mesozoic mammalian world of extraordinary complexity and diversity. The story of this fossil revolution is itself a remarkable one. Beginning in the 1990s and accelerating through the 2000s and 2010s, paleontologists working in the early Cretaceous rocks of northeastern China, the Yixian and Jiufadeng formations of Liaoning province, began uncovering exquisitely preserved fossils of early mammals. These fossils were remarkable not just for their exceptional preservation. Many preserved not just bones but fur impressions, soft tissue outlines, and stomach contents, but for the astonishing diversity of body plans they revealed. One of the first truly shocking discoveries was Rypenomammus, a mammal from the early Cretaceous of China that was, by the standards of Mesozoic mammals, enormous. Rypenomammus robustus was roughly the size of a large cat, and Rypenomammus giganticus was even larger, potentially reaching over a meter in length and weighing several kilograms. These were not the tiny, mouse-like mammals of the old textbooks. These were substantial animals, the size of a living opossum or a small badger. But the truly electrifying thing about Rypenomammus was not its size. It was what was found inside one of the specimens. Preserved in the rib cage of one Rypenomammus fossil, preserved as actual bones in what was clearly the stomach region, were the skeletal remains of a young dinosaur. A baby Psittacosaurus, a small ceratopsian dinosaur that is common in the same fossil beds, had been eaten by this mammal and was preserved in the process of being digested. The implications were staggering. Here was direct, unambiguous evidence that at least some Mesozoic mammals were not small, passive insectivores hiding from the dinosaurs. Here was a mammal that was eating dinosaurs, actively hunting or scavenging juvenile dinosaurs and consuming them. The relationship between Mesozoic mammals and dinosaurs was not simply one of tiny prey hiding from giant predators. It was, at least in some cases, the reverse, but Rypenomammus was just the beginning. The Chinese fossil beds and other fossil localities around the world continued to produce surprises that overturned every assumption about Mesozoic mammalian ecology. Volaticotherium antiquum, a gliding mammal from the Middle Jurassic of China, revealed that Mesozoic mammals had already evolved the ability to glide between trees, analogous to the modern flying squirrel, at a time when most people assumed they were all firmly terrestrial. Volaticotherium had a flight membrane, a patagium, stretching between its limbs, supported by a specialized skeletal structure. It was gliding through Jurassic forests over 160 million years ago, fully 40 million years before the evolution of any gliding placental mammal. Castorocota lutrosimiles, its Latin name means beaver-tailed, otter-like, was a Jurassic mammal from China that was adapted for semi-aquatic life. It had a broad, flat tail like a beaver. Its limbs showed adaptations for swimming. Its teeth were adapted for catching fish. It was, in the Middle Jurassic, a mammal filling an ecological role analogous to the modern otter or mink, a role that everyone had assumed was impossible for Mesozoic mammals to occupy. It weighed over 500 grams, making it another large for its time Mesozoic mammal that defied the old assumptions about uniform tiny body size. Fruitifossor winchefeli from the late Jurassic of North America was even more surprising. This animal had powerful digging forelimbs and a body plan remarkably similar to that of modern armadillos. It was a fossorial specialist, a digger, adapted for excavating the nests of colonial insects. Its teeth showed specific adaptations for eating termites or similar insects. Here was a Mesozoic mammal that had independently evolved a body plan strikingly convergent with modern Myrmecophagous, ant, and termite-eating mammals. A clear demonstration that the ecological experimentation of Mesozoic mammals was far more extensive than anyone had imagined. Juramaea sinensis, known from the late Jurassic of China, pushed back the origin of placental mammals to at least 160 million years ago, earlier than previously known. And Eomea scansauria, also from China, showed a placental mammal from the early Cretaceous with adaptations for climbing, grasping feet, a flexible body, limb proportions suited to moving through trees and shrubs. The picture that emerges from all of these discoveries is one of remarkable ecological diversity. Mesozoic mammals were not a single, homogeneous ecotype. They were a diverse radiation of animals, occupying a wide range of ecological niches, gliding, swimming, burrowing, climbing, predating, insectivore-ing, fish-eating, in body sizes that ranged from mouse-sized to cat-sized. They were, in other words, doing what mammals always do, diversifying into available ecological space with evolutionary creativity and biological flexibility. There is another important dimension to Mesozoic mammalian diversity that deserves extended discussion: the extraordinary variety of mammalian groups that existed during this period, many of which left no living descendants. Today, the living mammals are divided into three main groups: the monotremes, the marsupials, and the placental mammals. But during the Mesozoic, the mammalian family tree was far more bushy and complex than this simple three-way division suggests. There were numerous other groups of mammals, groups that were diverse and successful in their time, but that did not survive to the present, whose evolutionary lines ended, sometimes in extinction events and sometimes simply in competitive displacement by other groups. The multituberculates were perhaps the most spectacularly successful of these extinct mammalian groups. They first appeared in the late Jurassic and persisted all the way to the Eocene, a duration of over 120 million years, making them one of the longest-lived mammalian groups in the history of the lineage. They were rodent-like animals, not actually related to rodents, but convergently similar in many ways, with characteristic teeth that had multiple rows of cusps. The multituberculate name refers to these multi-cusped teeth. They were herbivores and omnivores, exploiting plant material and seeds in ways that their contemporaries largely could not. The multituberculates were enormously diverse. They ranged in size from mouse-sized to marmot-sized. They occupied a variety of ecological niches. Some were ground-dwelling, some were arboreal, some were semi-fossorial. They were globally distributed, found on every continent where Mesozoic and Paleogene fossil sites exist. And they survived the end Cretaceous extinction, recovering and diversifying in the Paleocene and Eocene alongside the placental and marsupial mammals. Their eventual extinction, sometime in the Eocene, roughly 35 to 40 million years ago, is generally attributed to competitive displacement by the rapidly diversifying rodents, which occupied similar ecological niches and apparently outcompeted the multituberculates in most regions. But for over 100 million years, the multituberculates were a major component of mammalian diversity, a reminder that the mammals we know today are only the survivors of a much larger and more complex evolutionary tree. There were also the Eutriconodonts, a group that included Rhipenomammus and several other interesting forms, and the Dimetrodonts, and various other lineages whose exact relationships to each other and to living mammals are still being worked out. The Mesozoic mammalian fauna was, in short, a rich and complex biological world that we are only beginning to understand in its full complexity. Let us now turn to a subject that is central to the story of Mesozoic mammals, but that is often overlooked in popular accounts: the question of mammalian metabolism and the ongoing refinement of the physiological systems that underpin the mammalian way of life. We established in Part 1 that the transition to endothermy, warm-bloodedness, was a gradual process that began in the Therapsid lineage long before true mammals appeared. By the time the first true mammals emerged in the late Triassic, they were almost certainly fully endothermic in the modern mammalian sense. But endothermy, while enormously advantageous in many ways, also imposes extraordinary energetic costs. A warm-blooded animal requires roughly 10 times as much food as a cold-blooded animal of the same body size, because it must continuously burn metabolic fuel to maintain its elevated body temperature. This enormous metabolic cost has driven the continuous refinement throughout mammalian evolution of the systems that acquire, process, and deliver energy to the body's cells. And during the Mesozoic, while the basic mammalian body plan was otherwise relatively constrained in body size by competitive exclusion from the dinosaurs, the internal physiology was undergoing continuous improvement. One critical system was the circulatory system. The mammalian heart is a 4-chambered pump that completely separates oxygenated blood from the lungs from deoxygenated blood from the body tissues. This complete separation allows blood to be delivered to the body's tissues at high pressure and with high oxygen content, fueling the high metabolic rates that endothermy requires. The evolution of this 4-chambered heart, shared with birds, though evolved independently, was a critical step in the development of sustained high-level endothermy. The respiratory system was also being refined. The mammalian lung, with its intricate branching structure of airways and its enormous surface area of alveoli, is one of the most efficient gas exchange organs in the vertebrate world. and critically, the evolution of the diaphragm, the muscular sheet separating the chest cavity from the abdominal cavity, gave mammals a breathing mechanism that is both powerful and efficient, capable of sustaining the high rates of oxygen delivery that an active, warm-blooded lifestyle requires. There's also the question of thermoregulation, the specific mechanisms by which mammals maintain their body temperature within the narrow range that allows their biochemistry to function optimally. Insulation, through fur or fat or both, is the primary passive mechanism, reducing heat loss from the body's surface. Active thermogenesis, the generation of heat through muscular activity and through specialized metabolic processes, is the active mechanism, allowing an animal to warm itself from within. The combination of insulation and active thermogenesis is what makes a mammal in the most physiological sense. The development of specialized heat-generating tissue, particularly brown adipose tissue, a type of fat that is adapted specifically for heat production rather than energy storage, is a distinctly mammalian innovation. Brown fat is rich in mitochondria and in a specific protein called uncoupling protein 1, which allows the mitochondria to generate heat directly rather than converting their energy into ATP. This tissue is particularly important in newborn mammals, whose small size makes them especially vulnerable to heat loss, and in small mammals that must maintain body temperature even in cold conditions with limited energy reserves. There is another dimension of Mesozoic mammalian biology that deserves careful attention: reproduction and parental care, and the ongoing evolution of the reproductive strategies that would eventually produce the extraordinary diversity of mammalian life histories we see today. We noted in Part 1 that the earliest true mammals were probably egg layers, like the modern monotremes. But during the Mesozoic, the two other major mammalian reproductive strategies, the marsupial and the placental approaches, evolved and began to diversify. And the differences between these approaches tell us something profound about the trade-offs inherent in mammalian reproduction and about the evolutionary pressures that shape the mammalian family. Marsupial reproduction is characterized by a very short gestation period. The young are born at an extremely early developmental stage, essentially as fetuses, and then continue their development externally, typically attached to a nipple inside the mother's pouch. The joey of a kangaroo at birth is roughly the size of a lima bean, blind, hairless, and with only its forelimbs developed enough to drag itself to the pouch. From this unpromising beginning, it develops over months into a fully functional young kangaroo, nourished by milk of changing composition as its developmental needs change. Placental reproduction, by contrast, involves a much longer internal gestation, during which the developing embryo is nourished through the placenta, a specialized organ that allows the exchange of nutrients and waste products between the mother's and embryo's bloodstreams without allowing the two bloodstreams to mix directly. The placenta allows placental mammals to give birth to much more developmentally advanced young than marsupials, young that, in many species, can stand and walk within hours of birth. Both strategies have advantages and disadvantages. The marsupial strategy requires less maternal investment before birth. The short gestation means the mother is less physiologically committed to any single reproductive attempt, and if conditions deteriorate, she can effectively abandon the joey with less cost than a placental mother could abandon a late-term fetus. The placental strategy, on the other hand, produces more developmentally advanced young that may have higher survival rates, and the placenta may allow more efficient transfer of nutrients and immune factors from mother to offspring. During the Mesozoic, both marsupials and placental mammals, or their close ancestors, were present and diversifying. The earliest unambiguous marsupials are known from the early Cretaceous. The earliest unambiguous placentals are known from roughly the same time, though the exact timing of placental origins is still debated. What is clear is that by the late Cretaceous, the final act of the dinosaur era, both groups were established and were diversifying alongside the multituberculates and other mammalian lineages in the shadow of the dinosaurs. The late Cretaceous is the final chapter of the mammalian Mesozoic experience, and it is a period of particular fascination. The world of the late Cretaceous, roughly 100 to 66 million years ago, was a world of extraordinary biological richness. The dinosaurs had reached the peak of their diversity. Flowering plants, the angiosperms, had evolved and were rapidly diversifying and spreading across the world's landscapes, transforming the nature of terrestrial ecosystems in ways that would have profound consequences for all the animals living in them. The rise of the flowering plants is one of the most consequential events in the history of terrestrial ecosystems, and its impact on the evolution of Mesozoic mammals deserves careful consideration. Before the angiosperms, the world's vegetation was dominated by gymnosperms, conifers, cycads, ginkgos, and their relatives, and by ferns and horsetails. These plants produce their seeds in relatively simple structures, and their fruits and seeds, while nutritious, are not produced in the extraordinary variety of forms that the angiosperms would later develop. The angiosperms evolved flowers, reproductive structures that attract animal pollinators, and fruits, nutritious structures that attract animal seed dispersers. This was the beginning of one of the most important ecological partnerships in the history of life, the mutualism between flowering plants and the animals that pollinate them and disperse their seeds. For the mammals, the rise of the angiosperms was enormously significant. The diversity of fruits, seeds, nectar, and pollen produced by flowering plants created a vast array of new food sources that had not existed before. Mammals with the right teeth and digestive systems could exploit these resources. The diversification of angiosperms during the late Cretaceous appears to have been accompanied by an increase in the diversity and ecological variety of the mammals living alongside them. Not a dramatic, sudden explosion, but a steady broadening of mammalian ecological and dietary diversity in response to the new food resources becoming available. The late Cretaceous mammalian fauna was not simply a collection of undifferentiated insectivores. There were mammals with teeth adapted for hard object feeding, crushing nuts and seeds. There were mammals with teeth adapted for processing plant material. There were mammals with teeth adapted for eating fruit. There were mammals adapted for different body sizes and different activity patterns. The diversification was not on the scale that would follow the end Cretaceous extinction, but it was real and it laid the groundwork for what was to come. And then came the asteroid. On a day 66 million years ago, a day that was, by all appearances, entirely ordinary up to that point, an object approximately 10 to 15 kilometers in diameter, traveling at roughly 20 kilometers per second, struck the shallow seas of what is now the Yucatan Peninsula. The impact released energy equivalent to roughly 100 million times the most powerful nuclear weapon ever detonated. In a fraction of a second, the impactor and everything in its immediate vicinity was vaporized. A shockwave of unimaginable violence propagated outward. A tsunami of colossal proportions swept across the ocean basins, but the immediate destruction, as horrific as it was not the primary killer. The primary killer was what happened to the atmosphere in the minutes, hours, days, and years that followed. The impact vaporized an enormous quantity of rock, both the impactor itself and the carbonate and sulfate rocks of the target site. This vaporized rock was ejected into the atmosphere and beyond. Some of it achieved suborbital trajectories, falling back through the atmosphere hours later as a global rain of hot ejecta that set fires across vast areas of the planet's surface. Soot from these fires was added to the dust and sulfate aerosols already filling the upper atmosphere. The result was a dramatic, prolonged reduction in the amount of sunlight reaching the Earth's surface. Temperatures dropped, photosynthesis was severely curtailed. The base of the food chain, the plants and phytoplankton that fix solar energy into organic compounds, was devastated, and without the base, the entire food chain above it began to collapse. The effects were not instantaneous. The biological crisis unfolded over weeks, months, and years as the environmental changes propagated through ecosystems. But the ultimate result was the end Cretaceous mass extinction, the elimination of roughly 75% of all species on Earth, including the non-avian dinosaurs, the pterosaurs, the marine reptiles, and vast numbers of marine invertebrates, plants, and other organisms. For the mammals, the end Cretaceous extinction was simultaneously the greatest catastrophe and the greatest opportunity in their evolutionary history. The catastrophe was real and severe. Many mammalian lineages that had existed through the Cretaceous were wiped out. The multituberculates were reduced but survived. Marsupials were severely reduced in diversity. Many other mammalian groups simply disappeared, but the placental mammals, or at least some of them, survived. And in surviving, they found themselves in a world that had been cleared of the competitive dominance that had constrained mammalian evolution for 165 million years. The dinosaurs were gone. The large ecological niches, the large herbivore roles, the large predator roles, the medium-sized browser roles, all of them were suddenly vacant. And the mammals were positioned, by virtue of their diverse biology and their flexible, warm-blooded, large-brained physiology, to fill them. What was it about the mammals that allowed them to survive the end Cretaceous extinction when the dinosaurs did not? This is one of the most frequently asked questions in paleontology, and the answers are complex and still partially debated. But but several factors stand out as likely important. Body size, again, was probably critical. The end Cretaceous extinction appears to have been particularly severe for large animals. Animals above a certain body size threshold were essentially wiped out. The non-avian dinosaurs, of course, included animals ranging from the size of a turkey to the size of a house, but they were disproportionately large relative to the surviving mammals. The mammals, constrained by competitive exclusion to mostly small body sizes during the Cretaceous, were inadvertently positioned in the safer size range when the extinction struck. Dietary flexibility was also likely important. The collapse of plant productivity following the impact would have been catastrophic for strict herbivores, animals that depended on living plant material for their food. Animals that could shift to seeds, to insects, to carrion, to any available food source rather than depending on a specific type that might no longer be available, would have had significant advantages. The mammalian jaw and its complex, differentiated teeth provided exactly this dietary flexibility. A small mammal that can switch from eating insects to eating seeds, to eating fungi, to eating small vertebrates as conditions change, is far better positioned to survive an ecological collapse than a large herbivore committed to a specific diet. Burrowing behavior may also have played a significant protective role. Mammals that lived in burrows would have been insulated from the worst of the temperature swings and the rain of hot ejecta that characterized the immediate aftermath of the impact. They would have had access to stored food resources, seeds and roots preserved in the soil, even when surface vegetation was destroyed. They would have been protected from the fires that swept across the landscape. The burrowing habit that had evolved in many mammalian lineages as an adaptation to the nocturnal, cryptic lifestyle of the Mesozoic became, in the context of the end Cretaceous disaster, a survival tool of critical importance. Warm blood endothermy may also have been advantageous in the cold, dark, resource-limited world of the immediate post-impact period. An animal that can maintain its body temperature independently of ambient temperature is better equipped to function in a world where temperatures are dropping and fluctuating wildly than an ectotherm that relies on environmental warmth to power its metabolism. And perhaps most importantly, though this is speculative, the mammals' complex brains, their behavioral flexibility, their capacity for learning and adaptation may have given them an edge in responding to rapidly changing conditions. The ability to modify behavior quickly in response to environmental change, to try new food sources, to exploit new microhabitats, to respond adaptively to novel conditions, is a function of the mammalian brain's expanded neocortex. In a world of sudden, catastrophic change, behavioral flexibility may have been the difference between survival and extinction. The world that followed the end Cretaceous extinction, the Paleocene epoch beginning 66 million years ago, was like the world that followed the end Permian extinction 252 million years earlier, a devastated, simplified, ecologically impoverished world struggling to rebuild. The forests were gone or dramatically reduced. The food chains were disrupted. The once complex ecosystems of the late Cretaceous had been reduced to a fraction of their former complexity, but the mammals were there, reduced, impoverished, their diversity temporarily diminished by the extinction event itself, but alive. And in the years and decades and centuries and millennia that followed, as the world slowly recovered, the mammals began to diversify in ways that were, by any standard, one of the most spectacular evolutionary radiations in the history of life on Earth. The Paleocene mammals were still, for the most part, relatively small and ecologically generalized. The immediate post-extinction world was not a simple cornucopia of opportunity. It was a damaged, stressed world with limited resources, and the animals exploiting it were still constrained by the limited food resources available. The forests were recovering, but slowly. The plant communities were reorganizing. The ecological niches were opening, but it took time for evolution to fill them. But evolution was filling them. With breathtaking speed on a geological timescale, the mammals were diversifying. Within 10 million years of the end Cretaceous extinction, an eye blink in geological time, the mammals had produced the ancestors of virtually all the major orders of placental mammals alive today. Ancestors of the rodents, ancestors of the bats, ancestors of the whales and their terrestrial relatives, ancestors of the elephants, ancestors of the primates. All of them evolved, or were already evolving, within the first 10 million years of the Paleocene. Let us follow some of these radiations in more detail, because they are extraordinary stories in their own right. Consider the whales. The evolution of the whales, the return of a fully terrestrial mammal lineage to the sea, is one of the most remarkable and well-documented evolutionary transitions in the fossil record. The ancestors of the whales were fully terrestrial mammals, hoofed animals related to the modern hippos, that began returning to an aquatic lifestyle sometime in the early Eocene, roughly 50 to 53 million years ago. Within roughly 15 million years of that initial return to the water, their descendants had become fully aquatic, with the hind limbs reduced to vestigial remnants, the forelimbs transformed into flippers, and the body streamlined into the fully oceanic form of the early toothed whales. The fossil record of this transition is now exquisitely documented. Pakistan, from the early Eocene of what is now Pakistan, was a four-legged, wolf-sized animal that was spending time in and near the water but was still capable of moving on land. Ambulocetus, slightly younger, was more committed to an aquatic lifestyle. Its hind limbs were already reduced, and its body was beginning to show adaptations for swimming, though it could still move on land. Rhodhostis from the Middle Eocene was largely aquatic, with greatly reduced hind limbs and a tail that may already have been developing the flukes that characterize modern whales. And Basilosaurus from the Late Eocene was a fully aquatic, elongated, serpentine animal, enormous in size, with vestigial hind limb bones that served no locomotory function, fully committed to a marine lifestyle. The entire transition from fully terrestrial, four-legged animal to fully aquatic whale occurred in roughly 15 million years. That is fast in evolutionary terms. It was driven by the extraordinary ecological opportunities of the post-Cretaceous ocean, where the marine reptiles, plesiosaurs, mosasaurs that had previously occupied the top predator niches had been wiped out by the end Cretaceous extinction, leaving those niches open for exploitation. Consider the bats. The evolution of flight in mammals, the evolution of the bats, is another remarkable story, though one where the fossil record is less complete than for the whales. The earliest known bats from the early Eocene, roughly 52 million years ago, were already fully formed flying animals. They had already evolved the elongated finger bones and wing membranes that characterize all living bats, and they were already using echolocation, the system of biological sonar that allows bats to navigate and hunt in complete darkness. We do not have fossil evidence of the intermediate stages between the non-flying ancestor of bats and the fully flying earliest bats. The transition appears to have occurred relatively rapidly in a lineage that did not leave a good fossil record. But the transition must have occurred, and it was presumably driven by the extraordinary advantages that flight offers to a small nocturnal mammal: the ability to exploit aerial insect populations that are otherwise inaccessible, the ability to travel rapidly between foraging areas, the ability to roost in locations, cave ceilings, high tree branches that are inaccessible to ground-based predators. The echolocation system of bats deserves particular attention because it is one of the most sophisticated sensory systems in the animal kingdom, and because its origins lie, once again, in the mammalian heritage of enhanced auditory capability that was refined during the nocturnal Mesozoic period. Bat echolocation works by emitting high-frequency sound pulses and listening to the echoes that return from objects in the environment. By analyzing the time delay, frequency shift, and pattern of these echoes, a bat's brain can construct a detailed three-dimensional representation of its surroundings, including the location, distance, size, shape, and even the surface texture of objects in real time while flying at speed in complete darkness. This is a biological computational feat of staggering complexity, and it requires a highly specialized auditory system and an enormously sophisticated neural processing apparatus. The mammalian ear, with its three middle ear ossicles, its specialized inner ear structures, is the foundation on which this system is built. The neural circuitry that processes echolocation information in the bat brain is the elaborated product of the same general mammalian auditory processing circuitry that was being refined in the early Mesozoic mammals as they navigated the nocturnal world. Consider the primates, our own order, the group to which we belong. The primates are the evolutionary family most immediately relevant to the story of human origins, and their early history is deeply intertwined with the post-Cretaceous mammalian radiation. The earliest primates appeared very shortly after the end Cretaceous extinction, possibly in the latest Cretaceous, more certainly in the early Paleocene. These earliest primates were small, nocturnal insectivorous animals, superficially similar in some ways to the modern tarsier or the small prosimians of Madagascar. But they carried, even in these early forms, the distinguishing features that mark the primate lineage: grasping hands and feet with opposable digits, forward-facing eyes giving binocular vision and depth perception, and compared to other mammals of similar body size, relatively large brains. The grasping hands and feet of primates are often interpreted as adaptations for life in the trees, for moving through a three-dimensional arboreal environment where the ability to grip branches securely is essential. The forward-facing eyes are interpreted as providing the precise depth perception needed to judge distances accurately when leaping between branches. These are the classic interpretations, and they have a great deal of supporting evidence. But there is also an alternative hypothesis, the visual predation hypothesis, which suggests that the primate features evolved primarily in the context of stalking and catching small, fast-moving prey, insects, and other arthropods in the fine branches of trees and shrubs. In this view, the grasping hands are adaptations for reaching out and seizing prey, and the forward-facing eyes are adaptations for the precise, binocular tracking of small, quick targets. Whatever the specific driving pressures, the result was a group of animals with an enhanced visual system, sophisticated manual dexterity, and large, complex brains, a combination that would, over the next 66 million years, lead to increasingly complex social behavior, language, technology, art, and ultimately science itself. The great mammalian radiation of the Paleocene and Eocene, the 60 to 55 million years following the end Cretaceous extinction, was not a smooth, orderly process of steady diversification. It was a complex, dynamic, sometimes chaotic process of radiation, extinction, competition, and replacement that played out over tens of millions of years in the context of a world that was itself undergoing dramatic environmental changes. The Eocene epoch, which followed the Paleocene, was characterized by extraordinarily warm global temperatures significantly higher than today, with tropical forests extending well into what are now temperate regions and minimal polar ice. This warm world was a world of extraordinary mammalian diversity, with an array of archaic mammalian orders, the condylarths, the creodonts, the euntathers, and many others that have no living descendants but that dominated the mammalian fauna of their time. And then the climate began to cool. Through the Eocene and into the Oligocene, global temperatures dropped significantly. The polar regions cooled, the tropical forests contracted. Grasslands, a new type of ecosystem dominated by grasses, the angiosperm family that would go on to cover enormous areas of the world's land surface, began to expand. The cooling climate and the spread of grasslands drove another major wave of mammalian evolution. Many of the archaic mammalian groups, the condylarths, the creodonts, were replaced by the more modern orders that now dominate the mammalian fauna. The rodents, the perissodactyls, horses, rhinos, tapirs, the artiodactyls, cattle, deer, pigs, camels and their relatives, the carnivores, dogs, cats, bears, weasels and their relatives, and others. The evolution of the horse family is one of the most celebrated and well-documented evolutionary sequences in all of paleontology. The earliest horses, animals like Eohippus, now more properly called Hyracotherium, were small, four-toed browsers from the early Eocene, about the size of a fox terrier, adapted for life in the forests that covered much of the world at that time. As the climate cooled and the forests gave way to grasslands, the horse lineage evolved larger body sizes, reduced toe numbers from 4 to 3 to 1, and more complex high-crowned teeth adapted for the harsh, silica-rich grasses that were replacing the softer forest vegetation. This trajectory from small, forest-dwelling browser to large, plains-dwelling grazer is a clear example of how mammalian evolution responds to large-scale environmental change. The mammals did not passively experience the opening of the grasslands. They evolved to exploit them, producing the extraordinary diversity of large grazing mammals that still characterizes many of the world's grassland ecosystems. The grasslands themselves deserve attention in this narrative because they represent one of the most important ecological developments in the entire Cenozoic era, the era of mammalian dominance that began 66 million years ago and continues to the present day. Grasses first evolved in the late Cretaceous, but they were a minor component of most Cretaceous ecosystems. In the Eocene, they began to expand. The Oligocene and Miocene, they became one of the dominant vegetation types across vast areas of the world, covering the interiors of continents with open, flat, windswept landscapes of extraordinary ecological productivity. Grasslands are harsh environments. The grasses themselves are difficult to eat. Their high silica content wears down teeth rapidly. They require specialized digestion, and the open, exposed landscape of a grassland offers little shelter from predators or the weather. But grasslands are also enormously productive. They can support much higher densities of large herbivorous mammals per unit area than forests can, because grass regrows quickly after being eaten, and because the open landscape allows the social grazing herds that are so characteristic of modern grassland ecosystems. The mammals that evolved to exploit the grasslands were transformed by the process. Their teeth became taller-crowned and more complex to withstand the abrasive silica of grass. Their digestive systems became more elaborate, with the evolution in many groups of rumination, the process of chewing already swallowed food, which allows the complex cellulose of plant cell walls to be broken down more thoroughly by microbial fermentation. Their legs became longer and their feet more specialized for running on hard ground at speed, as the open grassland landscape imposed new selective pressure for locomotory efficiency and escape speed. And their social behavior became more elaborate. The open grassland, with its excellent visibility and its absence of cover, is an environment where social living, the ability to detect and respond to predators collectively, to coordinate movement across large distances, to communicate effectively across open ground, pays enormous dividends. The great herds of the African savanna, the massive bison herds of the North American plains before the modern period, these are the products of millions of years of coevolution between the grassland ecosystem and the mammals that exploited it. Let us now focus on the primates specifically, following their evolutionary trajectory from those early Paleocene insectivores to the forms that would eventually lead to the human lineage. Through the Eocene, the primates diversified into a remarkable array of forms. The major split between the two main groups of living primates, the strepsirrhines, lemurs, lorises, and their relatives, and the haplorhines, tarsiers, monkeys, apes, and humans, appears to have occurred early in primate evolution, possibly in the Eocene or even earlier. The haplorhines, the group that includes our own ancestry, were characterized by further refinements of the primate visual system. The shift toward a more diurnal lifestyle in many haplorhines was accompanied by improvements in color vision. The ray evolution of trichromacy, three-color vision, from the dichromatic baseline that the nocturnal bottleneck had left. The fovea, a specialized region of the retina with the highest density of cone cells, providing the sharpest possible central vision, became more pronounced in haplorhines than in strepsirrhines, reflecting the increasing importance of detailed, high-resolution vision. In the Oligocene and Miocene, the apes, the hominoids, evolved from a monkey-like ancestor. The earliest apes appeared in Africa roughly 25 million years ago, and they diversified into a wide range of forms during the Miocene, a period sometimes called the Golden Age of Apes, when there were many more ape species than exist today, and they were distributed across a much wider geographic range, extending into what is now Europe and Asia. The apes differed from monkeys in several important ways. They lost the tail. Their bodies became larger and more upright in posture. Their brains became larger relative to body size. Their life histories became slower, longer periods of development and learning, longer reproductive intervals, longer lifespans. These changes reflected a shift in evolutionary strategy. Rather than producing many offspring quickly, the apes were investing heavily in fewer, more developmentally elaborate offspring, equipping them with the learned behavioral repertoire they would need to navigate complex social environments. The story of how one particular African ape lineage gave rise to the human lineage begins in the late Miocene, roughly 7 to 8 million years ago. The exact timing and circumstances of the human-chimpanzee divergence, the moment when the lineage leading to modern humans separated from the lineage leading to modern chimpanzees, is still a subject of active research and some debate, but the broad picture is clear. In the forests and forest margins of Africa, a population of apes, our ancestors, began to make a transition that would eventually transform the biological world. They began to walk upright on two legs. The evolution of bipedalism, habitual, obligate walking on two legs, is one of the most distinctive and consequential features of the human lineage. It freed the hands from locomotory function, allowing them to be used for manipulation, for tool use, for carrying food and offspring. It was energetically efficient over long distances on open ground. It allowed a new kind of long-distance travel across the increasingly open landscapes of late Miocene and Pliocene Africa as the forests contracted and the savannas expanded. The earliest members of our genus, the Australopithecines, a group that includes famous specimens like Lucy Australopithecus afarensis, were small-brained, upright walking apes that lived in the late Pliocene, roughly 4 to 2 million years ago. Their brains were not dramatically larger than those of chimpanzees. They used simple stone tools if they used them at all, but they were bipedal, they were upright, they were in the most important sense already on the trajectory that would lead to human beings, and then the brain began to expand. The expansion of the human brain, the dramatic increase in cranial volume that occurred in the genus Homo over the past 2 million years, is one of the most extraordinary events in the history of the mammalian lineage. It happened fast on an evolutionary timescale. The brain of Homo habilis, the earliest member of our genus, was roughly 600 cubic centimeters, about 1.5 times the size of a chimpanzee brain. The brain of Homo erectus, emerging about 2 million years ago, was larger still, up to 900 cubic centimeters. The brain of Homo heidelbergensis and early Homo sapiens was approaching modern human size, 1,200 to 1,500 cubic centimeters, roughly 3 times the size of a chimpanzee brain. This tripling of brain volume in just 2 million years, 2 million years being a very short time in the context of the 300 million year story we have been telling, is unprecedented in the vertebrate world. No other lineage has expanded its brain so rapidly, so dramatically, and with such far-reaching consequences. What drove this brain expansion? This is one of the most actively debated questions in human evolutionary biology, and a full discussion would require a documentary of its own, but the broad outlines are clear. The social complexity of the human lineage, the demands of navigating an increasingly complex social world of alliances, rivalries, cooperation, and competition, placed enormous selective pressure on cognitive capacity. The use of tools which required planning, foresight, and the ability to learn from others rewarded increased brain power. The development of language, the ability to communicate complex, abstract information symbolically, both required and drove further brain expansion. The result is the human brain, the most complex object as far as we know in the observable universe. 100 billion neurons, each connected to thousands of others through synapses whose total number exceeds the number of stars in the Milky Way. Capable of language, mathematics, music, art, philosophy, science. Capable of asking where it came from, and, as we are doing right now, of finding the answer in the deep past. The brain that allows you to watch and understand this documentary, that allows you to process language, to form mental images of ancient creatures, to feel the weight and wonder of deep time, that brain is the culmination of over 300 million years of synapsid evolution. It is the end product of every transformation we have traced in this documentary. The temporal fenestra of the earliest synapsids, the complex teeth of the therapsids, the upright posture of the cynodonts, the migrated ear bones of the earliest mammals, the enhanced olfactory system of the Mesozoic nocturnal mammals, the forward-facing eyes of the earliest primates, the expanding neocortex of the apes. Every one of those innovations, built upon the one before, contributing to the next, accumulating over 300 million years in an unbroken chain of inheritance and refinement. Nothing in this history was designed, nothing was inevitable. Each innovation was the product of natural selection acting on random variation, favoring the variants that survived and reproduced better than their competitors in the specific environmental context of their specific moment in time. And yet the trajectory, looked at across its full 300 million year span, has a kind of directionality, not because it was heading anywhere in particular, not because there was a destination in mind, but because the features that proved advantageous in one context tended to create new possibilities that were advantageous in the next context. Warm blood made possible sustained activity. Sustained activity made possible the exploitation of more demanding, higher quality food sources. Higher quality food sources made possible larger brains. Larger brains made possible complex behavior. Complex behavior made possible language and culture. Language and culture made possible the cumulative transmission of knowledge across generations, and it is that cumulative transmission of knowledge that distinguishes the human lineage most profoundly from all the others. There is a theme that runs throughout this entire 300 million year story, and it deserves to be named explicitly as we approach the conclusion of our documentary. That theme is survival through catastrophe. The synapsid lineage did not reach its current apex by following a smooth, uninterrupted path of progressive improvement. It reached it by surviving catastrophes that destroyed everything else. The end Permian extinction nearly annihilated the lineage. The end Triassic extinction further constrained it. The end Cretaceous extinction killed off many of the mammalian groups that had diversified through the Mesozoic. Even within human evolutionary history, there is evidence of severe population bottlenecks, events that reduced the effective population size of early human ancestors to alarmingly small numbers. Each of these catastrophes was, in the moment, a tragedy. The end Permian extinction destroyed a world of extraordinary beauty and complexity, the world of the gorgonopsians and the dicynodonts, and the myriad other Permian organisms that we will never fully know. The end Cretaceous extinction destroyed the dinosaur world, a world that had been developing for 165 million years and that contained animals of staggering variety and grandeur. But each catastrophe was also, in the long run, a filter, a brutal, arbitrary, enormously costly filter that happened, through pure contingency, to spare the ancestors of the mammals. And with each pass through the filter, the surviving lineage found itself in a world where the competitive pressures that had previously constrained it were suddenly removed, where ecological space was open, where new opportunities had been created by the very catastrophe that had nearly destroyed everything. This pattern, catastrophe creating opportunity, is one of the deepest patterns in evolutionary history. It is not a comfortable pattern. It is not a reassuring pattern. It suggests that the richness and complexity of the modern biological world, including the remarkable, unprecedented complexity of the human mind, is built on a foundation of repeated massive destruction. That we are here because of extinctions. That our existence is inseparable from the endings that preceded it. Let us now draw together all the threads of our story and see the full picture that they make. 312 million years ago, the amniote lineage split. On one branch were the sauropsids, the ancestors of the reptiles and the birds. On the other branch were the synapsids, the ancestors of the mammals and of us. The synapsids diversified through the Carboniferous and into the Permian, producing the pelycosaurs and then the therapsids, animals with increasingly complex teeth, increasingly upright postures, increasingly high metabolic rates. They became the dominant terrestrial vertebrates of the Permian, filling every major ecological role in terrestrial ecosystems across the supercontinent of Pangaea. 252 million years ago, the end Permian extinction nearly destroyed them, destroying the gorgonopsians, reducing the dicynodonts to a handful of survivors, leaving the cynodonts as the primary synapsid survivors of the catastrophe. Those cynodont survivors carried forward the biological innovations that define the synapsid lineage, and they continued to refine them through the Triassic, even as the dinosaurs rose to dominance and the synapsids were pushed into the ecological margins. By the late Triassic, true mammals had emerged from the cynodont lineage, small, warm-blooded, sharp-eared, large-brained relative to their body size, possessing the uniquely mammalian dentition and the three-ossicle middle ear. Through the 165 million years of the dinosaurs' dominance, these early mammals lived in the nocturnal margins, refining their sensory systems, developing new reproductive strategies, diversifying into a wider range of ecological roles than anyone previously imagined. 66 million years ago, the end Cretaceous impact cleared the stage. The mammals survived by virtue of their small size, their dietary flexibility, their burrowing habits, their metabolic resilience, and then they radiated into the ecological vacuum left by the dinosaurs' extinction. Within 10 million years, the ancestors of virtually all living mammalian orders had appeared. Within 30 million years, the great mammals of the modern world, the whales, the bats, the horses, the elephants, the primates, were well established and diversifying. 5 to 7 million years ago, within the primate lineage, the human lineage began its divergence from the ape ancestor shared with chimpanzees. Bipedalism evolved. Brain size began its extraordinary, unprecedented expansion. Language emerged, culture accumulated, and eventually, after a journey of over 300 million years that began with a small hole in the skull of an ancient amniote, we arrived here at the capacity to tell this story. There is one more thing that deserves to be said before we close this documentary, something that goes beyond the purely scientific story of mammalian evolution and touches on what this story means for how we see ourselves. We live in a culture that tends to see human beings as special, as apart from nature, as the pinnacle of a purposeful creation, as fundamentally different in kind from the other animals with which we share the planet. These are understandable tendencies. The human mind is genuinely extraordinary. The capacity for language, for abstract thought, for science and art and music and philosophy, these are things that no other animal possesses to anything like the same degree. We are remarkable, but we are not apart. We are not fundamentally different in kind from the other mammals. We are deeply, intimately, biologically continuous with them. The warm blood that keeps our bodies at a constant temperature is the same warm blood that warms a dog or a whale or a bat. The milk with which human mothers nourish their infants is a direct biological descendant of the secretions with which a Triassic cynodont mother nourished her hatchlings. The 3 tiny bones in our middle ear that allow us to hear music and speech and birdsong were, 250 million years ago, part of the jaw joint of our distant ancestors. We are made of inherited biology, every bit as much as we are made of stardust. We carry within our bodies the legacy of over 300 million years of evolution, the accumulated wisdom, in a sense, of every surviving ancestor in that unbroken chain. The fear that constricts our chest in a moment of danger is the legacy of small mammals that survived in a world of giant predators. The comfort we feel in the presence of familiar faces and trusted companions is the legacy of the social bonding that mammalian parental care made possible. The restlessness that drives us to explore, to learn, to understand, that insatiable intellectual curiosity that has produced every great discovery in human history, that is the legacy of the expanding primate brain, refined by millions of years of selection for navigating complex social worlds and solving novel ecological problems. Understanding where we come from does not diminish us. It reveals the extraordinary depth and richness of the biological heritage that we carry, reveals the improbable and fragile chain of contingencies that led from an ancient carboniferous amniote to a modern human being, reveals the connection between our existence and the existence of every other mammal alive today, our cousins across the 300 million year family tree. The story we have told in this documentary is, in its broadest outline, a story of persistence. Persistence through the end Permian extinction, which nearly erased everything. Persistence through the long Mesozoic in the margins and the shadows as the dinosaurs ruled the world. Persistence through the end Cretaceous disaster, which killed 3/4 of all species. Persistence through the cooling of the Eocene and the radical environmental changes of the Miocene and the Pleistocene. At every stage, the lineage could have failed. At every stage, there were moments when it nearly did. At every stage, some small number of survivors carried the torch forward, not heroically, not consciously, not with any awareness of the magnitude of what they were preserving, but simply by surviving, by finding enough food, by avoiding enough predators, by staying warm enough and small enough and flexible enough to make it through one more crisis, one more bottleneck, one more catastrophe. And here we are, 312 million years after that first split in the amniote lineage. 66 million years after the last dinosaur that was not a bird drew its last breath. 7 million years after the human lineage began its divergence from the ape ancestors we share with chimpanzees. 300,000 years after anatomically modern humans first appeared. Here we are carrying in our bodies and brains the accumulated inheritance of all of that, building cities, composing symphonies, launching spacecraft, writing poetry, and making documentaries about where we came from. The dinosaurs were magnificent. They ruled the world for 165 million years with a biological power and ecological dominance that nothing before or since has matched in its duration. But they are gone. They survived as birds, beautiful, diverse, extraordinary in their own right. But the non-avian dinosaurs, the great reptilian lords of the Mesozoic world, are fossils. The synapsids are not fossils. The synapsids are alive. They are the whale breaching in the deep ocean. They are the elephant mourning its dead with a tenderness that looks, to human eyes, remarkably like grief. They are the bat threading the night air by the miraculous instrument of its own voice. They are the dog pressing its warm flank against the leg of its companion. They are the mother nursing her infant. They are the child asking why. They are you. This has been the story of the true origin of mammals, a story that began before the dinosaurs, survived the worst catastrophe in the history of life, endured 165 million years in the shadows, and ultimately produced the most complex minds that evolution has ever fashioned. It is a story of unimaginable duration and extraordinary contingency. It is a story of survival against all odds, of biological innovation accumulated over geological time, of a lineage that refused, not consciously, not heroically, but simply biologically, persistently, tenaciously, to disappear. It is your story, and now you know it. This has been Before the Dinosaurs, the true origin story of mammals. Thank you for watching. Please share this documentary with anyone who has ever wondered where we truly come from. The answer reaches much further back than most people imagine, and it is a story worth knowing in its full, magnificent depth. If you found this journey through deep time as extraordinary as we did in making it, please leave a comment below telling us where you watched and what moved you most. Your engagement with this history keeps these stories alive. Subscribe for more journeys into the deep past. There is no shortage of extraordinary stories waiting to be told. Take a moment, just a moment, to think about what you are. You are warm, you breathe steadily. Your heart beats in a rhythm that has been refined over hundreds of millions of years. You have a jaw capable of chewing, ears capable of hearing the finest whispers, and a brain wrapped in layers of complexity that no other creature on this planet can match. You think, you remember, you dream. And you may have always assumed, as most people do, that the story of creatures like you, mammals, began sometime after the dinosaurs disappeared, roughly 66 million years ago, when the dust settled and a few small, frightened animals crept out of the rubble to inherit an empty world. That assumption is wrong. Beautifully, magnificently, breathtakingly wrong. The real story of mammals, the real story of you, begins not 66 million years ago, but over 300 million years ago, in a world so ancient and so alien that the very continents looked nothing like they do today. It begins before the first dinosaur ever drew breath. It begins in a period of Earth's history that most people have never heard of, in creatures that most people have never imagined, in an evolutionary drama so vast, so violent, and so extraordinary that the rise of the dinosaurs, for all its fame and cultural glory, is almost a footnote by comparison. This is the story of the synapsids. This is the story of the lineage that survived everything: volcanic apocalypse, the near-total annihilation of complex life, 200 million years of living in the shadow of giants, and persisted, refined itself, and ultimately produced every mammal alive today, including the one watching this video right now. This is your true origin story. Please let us know in the comments where you are watching this history video and what time you are watching it. And be sure to subscribe to the channel for future videos. Let's start at the very beginning, not the beginning of the universe, not even the beginning of life, but the beginning of the chapter that matters most to this particular story. To do that, we need to understand the world as it existed roughly 310 million years ago, deep in a period geologists call the Carboniferous. The Carboniferous world was extraordinary by any measure. The planet was dominated by vast, swampy forests, enormous stands of towering club mosses and tree ferns that stretched across vast stretches of the landmasses. These plants were pumping oxygen into the atmosphere at a rate that the world had never seen before and would not see again, producing an atmosphere so rich in oxygen that insects could grow to staggering sizes. Dragonflies with wingspans of over 2 feet patrolled the skies. Millipedes that stretched nearly 8 feet in length crawled across the forest floors. The air was thick, warm, and extraordinarily productive. It was in this environment, this lush, oxygen-saturated, biologically explosive world, that a critical transition in vertebrate history was quietly taking shape. Vertebrates, creatures with backbones, had already made the long journey from the seas onto land. Fish had developed limbs and the ability to breathe air. The first amphibians had colonized the terrestrial environment, and then, from within that amphibian stock, something remarkable had happened. A group of animals had developed the amniotic egg, an egg with a protective membrane inside that prevented it from drying out, that allowed the embryo to develop in its own private pool of fluid even on dry land. This was revolutionary. This was the key that unlocked the terrestrial world in ways that had never been possible before. The animals that possessed this amniotic egg were called amniotes, and they were the ancestors of everything that would come after: every reptile, every dinosaur, every bird, every mammal. And very early in the history of amniotes, something crucial happened. The amniotes split. They divided into 2 distinct evolutionary branches. This split, one of the most consequential events in the history of life on land, occurred roughly 312 million years ago. And understanding it is absolutely essential to understanding everything that follows. On one side of this split were the creatures that would eventually become reptiles and, later, dinosaurs and birds. These are the sauropsids, the lizard-faced ones. Their lineage would go on to produce some of the most iconic animals in the history of the planet. On the other side of this split, and this is the critical point, were the creatures that would eventually become us. These are the synapsids, the fused arch ones, named for a particular feature of their skull structure. And understanding what made synapsids different from the very beginning is the foundation of everything this documentary is about. Because here is the truth that so many people miss: mammals did not evolve from reptiles. This is one of the most persistent misconceptions in popular science, and it deserves to be dismantled clearly and definitively. The synapsid lineage is not a branch that broke off from the reptile family tree. Synapsids and reptiles are siblings, not parent and child. They share a common ancestor, yes, the early amniotes of the Carboniferous, but they diverged from each other over 300 million years ago, and they have been traveling their own separate evolutionary paths ever since. Synapsids did not become reptiles first and then become mammals. They were always something distinct. They were always on the path that would lead, eventually, to warm blood, to live birth, to nursing young with milk, to complex teeth, to large brains, to language, to consciousness itself. Every one of those traits has deep, ancient roots in the synapsid lineage, and those roots were already growing in a world that existed long before the first dinosaur took its first step. this is a story of extraordinary duration. We are talking about a lineage that has been refining itself, adapting, surviving catastrophe, and persisting for over 300 million years. To put that in perspective, the dinosaurs existed as a group for roughly 165 million years. The synapsid lineage has been going for nearly twice that long, and it is still going. Every mammal alive today, from the